Okay, Bismillahirrahmanirrahim. Assalamu alaikum and good day, ladies and gentlemen. Welcome back to the second day of 8th NRAB Comstech Regional Seminar for the year 2025. Theme of the seminar is plant materials and their clinical validation and management of diabetes and metabolic disorders, challenges and scope. So for the first technical session of this day will be chaired by Professor Dr. Nassar Ahmed. Professor Dr. Nassar in himself is an accomplished scientist. He is right now affiliated as a reader in clinical biochemistry, Department of Life Sciences, Manchester Metropolitan University, United Kingdom. So I'll request all of you to please welcome Professor Dr. Nassar as a chair. And now I'll hand over the mic to Professor to call upon our the very first speaker of the technical session. Over to you.
Okay, first of all, good morning to all of you. Assalamu alaikum. I'm hoping that you're actually enjoying this lovely seminar. We're on the second day of this seminar and it's session 3. We have 3 speakers, so I'm going to introduce the first speaker, who is Professor Barry Noller. He is Principal Research Fellow, Centre for Mind-Land Rehabilitation, University of Queensland in Australia. He has a long history with NRAP from its inception. He's also a board member. I've got to know him a bit since yesterday, and he's also a wonderful, wonderful human being. So over to Professor Noller.
Thank you, Chair. And it's been good to meet you also and chat informally. And I'm also very pleased to be here for yet another ANRAT event. In the tradition of following many of these workshops over many years, and I remember today also my esteemed colleague, Professor Mozi Husman Zaman, who I would love to have seen again here, my great friend. So my talk today derives from my work in Australia, and briefly, I have a co-author, Fiona Henderson, who was a master's student, and her interest was has ended up being in space science, and after finishing her study with me, she got a scholarship to Oxford, and today she's studying water on the moon. My talk here is about water on Earth and the effect of salinity composition on freshwater aquatic species in river water. So in Australia, like other dry locations in the world, the regular salinity composition is a significant issue affecting freshwater systems, especially in catchments that are highly modified, in this case by coal mining activity. Salinity as part comes from geological sources. Salinity in rivers can vary in composition due to changes in aquifer lithology, natural salt deposits in landscape, and salt accumulation due to land altering activities, mining, agriculture, and clearing. So monitoring waterway salinity is vital to managing adverse impacts on agriculture, drinking water supplies, and ecosystem health. Excess salinity is a significant issue in the regions in Australia, Murray-Darling Basin, the Hunter River, New South Wales, and also in Victoria and Southwest Australia. The focus here is on the Hunter River Valley case study from the Upper Goulburn River catchment within the catchment of the Hunter River to characterise different salinity associated with mining and natural geogenic mineralisation. So the aim study identify how salinity composition influences river chemistry and ecosystem health, and then to demonstrate that salinity factor approach is a valid method to characterize different sources and compositions of salinity and in river water. So the study focus is the Hunter River Valley in New South Wales, shown there, which is a very large coal mining area, even on the global scale. And within the Hunter River Valley itself, the geology is dominated by sedimentary Permian sequences. And in the central area, where there's a large blue colour, is associated with extreme levels of salinity derived from the coal mineralisation, which will achieve up to 7,500 microsiemens per centimetre of salinity in the river water, just from natural sources. Mining tends to expose that, and then discharges of water increase salinity. So this catchment has a dominating effect arising from salinity and its release. So the catchment is has mixed land use of covering agricultural activities, livestock production, and extensive coal mining. There's also a significant grape growing industry there, and other boutique agricultural activities, plus other urban developments. And the chain of these are effectively railway lines that go from the coal mine down to the coast. So there are two on the left-hand side there called Moolarben and Wilpinjong that I'll refer to in the upper Goulburn River catchment. And all of these mines, and there are many of them, export coal even today through the port facility of Newcastle at the estuary of the Hunter River. Excuse me. So salinity coefficient, which is Ke, is the ratio of total dissolved solids in milligrams per litre to electrical conductivity, and is utilised to distinguish and compare salinities in a catchment. Long-term water quality data is retrieved from various government databases. These are quite extensive. A map of Ke distribution and magnitude of total dissolved solids versus EC, when plotted as a cumulative distribution, shows the influence of geologic mineralization on the actual values of salinity, which come from its natural sources and the arising from the various mixtures of cations and anions. And when we look at a plot of electrical conductivity to total dissolved solids, we find a relationship which is almost linear. And in here, there's designated one of the aquatic test species, which was collected in the wild, mayfly larvae, is shown to arise from a tributary, a sub-tributary, which came out of a granite sequence where there was a large population of mayflies, but I'll come back to that later. When we look at the cumulative distribution of the salinity conversion factor, it separates out differences, which indicates that there are significant variations in the composition of the salinity itself. When we look at it, at the plot and this form, normally this feature is not displayed. So the cumulative distribution gives access to this variation. So we see on the upper left side there that there's diamonds there are associated with water coming from coal mines. So there's some similarity there. And the other variations are waters that come out of different geological materials, including coming out of granite, which is much cleaner. So comparison with the aquatic toxicity thresholds for salinity with a range of aquatic freshwater species follows Australian water quality guidelines and has been used to undertake by developing to develop a site-specific guideline for salinity and the other conditions. This is for ultimately for regulatory purposes. Aquatic toxicity data with test freshwater aquatic species is supported by other data for species abundances and to help interpret the significance of variations in toxicity in water which is derived from salinity itself. This approach is site-specific and reveals that ecotoxicity risks from treated wastewater from coal mining discharged in the upper Goulburn River cat of the Hunter River can be controlled and provide satisfactory environmental protection. The ecotoxicity work was done by a commercial laboratory in Sydney, which has a very specific, gives very specific expertise in undertaking these complex aquatic ecotoxicity tests. So the Australian Guidelines, ANZG 2018, follows a decision tree process, which allows in the final step to use aquatic toxicity testing to determine the likely effects of the water constituents on local aquatic species. Here we have the formula: total dissolved solids is equal to K, the factor, times EC. So the factor may vary, as we see from the cumulative distribution, and that's the cumulative plots allow, as I said, access to understanding how these variations, when they occur. So with aquatic species used in the sensitivity, the data is brought together in what's called the site sensitive distribution, species sensitive distribution, which uses chronic and aquatic and acute responses of organisms. And then there's a complex program called Burr Type 3 statistical distribution, which brings together all the toxicity responses of a range of different aquatic species. And in this work, 8 different species are used, covering a range of different trophic levels, and were all generally available by the ecotoxicity testing laboratory, except for the mayfly larvae, which the government regulators asked to be used in the testing, and they had to be collected in the field, and hence the designation of their habitat, which was in granite as opposed to Permian sedimentary feature, showed that they preferentially lived in a habitat where the water had very low salinity and therefore thrived there unaffected. So, but they were collected successfully and taken to the aquatic toxicity laboratory by road, kept cool and used in the excuse me, in the toxicity testing. So the Burr 3 distribution uses the 10 percentile toxicity of the range of species, and then puts them into a statistical species sensitivity distribution, and enables, in this case for sulfate, to give the 95 percentile value for the distribution of all these species, which on this plot you can see is 819 milligrams per litre with a confidence interval. And it's a very neat, sophisticated way to display the relative sensitivity of the range of aquatic species from being very sensitive to less sensitive, and is a very neat application. However, I mean, this kind of testing for 8 species costs something like $70,000 to undertake. So it's not a trivial exercise, but it's very effective in terms of enabling a site-specific guideline to be developed. So the sulfate concentration is converted by actual measurements to electrical conductivity and also ultimately to salinity using a site, a location-specific K value and not a generalized value. So at this coal mine, Moolarben, it was the default was of K was 0.73 as measured, not an average figure. So the although the direct ecotoxicity testing calculated for site-specific guideline value for EC, electric conductivity for 95% aquatic species protection of 2,649 microcents per centimetre. Background concentrations were also considered with the proposed recommendation details following the guidance of the Australian guideline method. Now, a second mine nearby, and this is about 2 years apart, gave a 95% confidence interval or mean value of 770 milligrams per litre of sulfate. Now, that compares with the 810 for the different mine, which is about 20 kilometres apart, but 2 years apart in time, showing that at different times there can be similar toxicity values at different locations. Normally that comparison is not made because of the relative costing of the testing at each site, but it shows how similar toxicity values are in a nearby different location. So all the process here is for both of these mines is effectively the same. So at this mine, Wilpinjong, so the ecotoxicity testing demonstrated no significant difference, significant toxicity on any of the aquatic test species. It was anticipated that test species would show a response to enable a site-specific guideline derivation. And in fact, to get site-specific values, there needed to be an addition of salt to the test solutions to induce toxicity, which happened. So the sulfate site-specific distribution for 95% protection, as I just showed, was 770 compared with 819 at Moolarben, the other site, and then various comparisons with the actual mine water, which was used for testing. So I said, which wasn't at an EC of 4,060 microcents per centimetre salinity, 2,904 was not sufficiently toxic to induce mortality of aquatic species without adding additional salinity, which is what was done in practice. So in conclusion, the salinity factors are demonstrated as an effective tool for salinity classification. Salinity is not just one species, although it's treated as such that, you know, there's just, it's just salinity itself. It covers a range of compositions. And so the factor is a way to discriminate with different sources. Site-specific species distribution can generate site-specific guidelines and give a better understanding of ecotoxicity risks within different catchments for salinity or expressed, based on concentration units or EC, electric conductivity itself. So ecotoxicity response helps be able to understand, but also how metal speciation is involved in the responses. Now, I'm not talked about different metals because there's not time for that, but metal speciation can be affected by higher alkalinity and also hardness within as part of the high salinity, and also be present in heavily river modified environments. So I want to thank you for listening to my talk, and if there's any time left, we might have a question. Thank you very much.
Okay, thank you for that lovely, clear presentation. Any questions from the audience? Can we get a speaker, please? Mike, sorry. Anyone with a mic?
Yeah, well, as part of the 8 species, I think there's 1 or 2 species of algae. They tend to be the most sensitive. The mayfly, as it turned out, larvae were between the middle and towards the least sensitive species. So, I mean, there's also lemna there, you know, like duckweed. So within the 8 species, I mean, there's apart from algae and the species that I've mentioned, there's also pedosirens, you know, micro zooplankton. So the intent is to not just use 1 species, but to have 8 species and then have the integrated you know, statistical analysis to show the combined effect of all species.
Because some algal species they just work as indicators, and they let you know that which content is more in water.
Oh, well, that's right, but they're part of the food chain, and that's a significant feature.
Thank you.
Any further questions? No? Okay, in that case, thank you very much, Professor Nola.
Thank you, everyone.
Right. Our next speaker is Professor Shamsun Nahar Khan. I know Shamsun when she was doing her PhD, with a mere PhD student. That was a good few years ago. She got her PhD in 2008, where she studied at the International Center for Chemical Biological Sciences, University of Karachi. 2010 to 2013, she did her postdoc at Harvard University in the USA in molecular and cellular biology. She's got an impressive list of publications, including one in Nature Communications that has an impact factor of 17.5, so very impressive indeed. She's got another 60 international peer-reviewed publications, 7 US patents, 7 book chapters, invited lectures at 30 conferences in USA, Europe, and Asia. 2025 onwards, she's been a member of the Bangladesh Food Safety Authority. 2016 to 2017, executive member of Global Young Academy in Germany. 2016 onward, young affiliate for TWAC, and 2016 onwards, profiles of women in science in Asia. More importantly, 2011, she was awarded the gold medal from the Bangladesh Academy of Sciences. And in 2012, she got TWAC's first prize in chemistry, and that was more specifically Professor Abar Rahman Prize. So over to Shamsun. She's talking about perspectives of new alpha glucosidase inhibitors as anti-diabetic drugs.
Assalamu alaikum. Very good morning to all of you. And thank you very much, Doctor, for your very kind introduction. He has been mentor since I'm student, so since 2002. Thank you very much. So today I'm going to talk about perspective of new alpha glucosidase inhibitors as anti-diabetic drugs. Yesterday we have observed several presentations, especially Professor Begum Rokeya, and she's also one of the wonderful person in the anti-diabetic medicinal plants. And there were several questions and remarks regarding the bioactive compounds. So here I think I'm presenting that's the reason the pure molecules which are having anti-diabetic properties, basically due to the alpha glucosidase inhibitors. Here I'm going to show the mechanism, the detailed kinetic studies, and also correlation with the three-dimensional quantitative structure activity relationships, because the consistent biological and chemical relationship is very important to develop any drugs, and also prediction for new molecules, so how to design or synthesize new molecules, and which definitely we want to have a more active and with less side effects. So currently I'm working as a professor at the East West University, Dhaka, since 2008. And now I'm serving as professor at the department. First of all, what is alpha-glucosidase? Alpha-glucosidase is an enzyme. It belongs to a hydrolysis classes, and it is responsible for the hydrolytic cleavage of carbon-oxygen, carbon-nitrogen, and carbon-carbon bonds. Yeah, we can see the 3D structures of different hydrolysis classes of enzyme, but importantly, alpha-glucosidase crystal structure has not been yet isolated or deposited in rcsb.org. So that is the PDB profiles of all the crystals proteins. Carbohydrate is a very complex molecule, an important nutrient of our food, which is being hydrolyzed and converted to smaller unit for digestion, basically the monosaccharide, which is responsible for the blood glucose enhancement of the blood glucose levels. Actually, alpha-glucosides act as a catalyst, increase the rate of reaction. As we can see in the diagram, this is the first graph is without an enzyme, and we can see the activation energy profile is quite high, and another one is with the enzyme, where the activation energy is low. So alpha-glucosidase increase the rate of reaction and lowering the activation energy of a reaction, and enzymes, as all the enzymes, speed up the reaction by a factor of many millions. Actually, enzyme produce the product by following alternative pathway or shortcut pathways. Alpha-glucosidase inhibitors, interestingly, are already present in the market. The structures of acarbose and voglibose and miglitol is presented here. And acarbose is manufactured by Bayer Pharmaceuticals, and the doses was 50 to 300 milligram, 2 to 3 times a daily. And miglitol is the manufacturer is Pharmacia. The doses is 50 to 150 milligram, 2 or 3 times a day. These drugs are already used by the clinicians, but there are several side effects, and among them is flatulence, uncomfortable after having these drugs actually stimulated us to take the challenge to have the new alpha-glucosidase inhibitors. There are several clinical importance of alpha-glucosidase inhibitions or inhibitors. As the diabetic patients have multiple complications, several of them are having HIV or AIDS and cancer and some other these clinical conditions. So the importance of alpha-glucosidase, along with the anti-diabetic agents, has antiviral agents or anti-tumor activity, anti-cancer agents, and anti-glycogen agents as well. So glucosidase inhibitors versus diabetes. Here I showed the pictures. The alpha-glucosidase present located in the small intestinal brush border of the human. The starch is major food component of our food component, and it's converted to oligosaccharide by pancreatic alpha amylase, and this oligosaccharide is further converted to glucose by membrane-bound alpha-glucosidase. And this free glucose can be only absorbed and enhance the blood glucose level. So here, if we can block this pancreatic alpha amylase or the membrane-bound alpha-glucosidase, then postprandial blood glucose levels can be controlled in case of type 2 diabetes. There are local amino acid sequence similarity observed between alpha amylase, mammal alpha amylase, and alpha-glucosidase. So definitely the inhibitors have the similar activities, and also we found acarbose also inhibit alpha-glucosidase in the yeast alpha-glucosidase and mammal alpha-glucosidase. The summary of the work, natural class of alpha-glucosidase, we reported in the studies: fluoroglucinol, iridoids, flavonoids, anthanoids, xanthones, acridones, physaline, triterpene, diterpene, benzene derivatives, long-chain fatty acids, steroids, lignin, which were identified as new and novel natural alpha-glucosidase inhibitors. Synthetic class of alpha-glucosidase inhibitors were reported in the studies was biscumarine, isocumarine, acidones, alkaloids, and both reserpine type of indole alkaloids, indole fused triterpene derivatives, organometallic complexes, for example, vanadium complexes, ruthenium complexes, and tin complexes, which were identified as new and novel synthetic inhibitors. I'm giving few of the structures activity relationships, SRR studies. Here we can see in case of compound 2, the methoxy group at 2' position is very crucial for the activity. If it is replaced by the hydroxyl group, in case of compound 3, the activity is fallen down to 1100 micromolar. And methoxy group at C4' position improved the activity further to 114 micromolar, which is 3.7 times more active than the standard deoxynitromycin, which is the standard was 425 micromolar. The compounds were isolated from Irish germanica, Melissia curari, and Irish tinifol. Similar effect also found in compound 7. Methoxy group at C1' position showed IC50 value 67 micromolar, which is 6.3 times more active than the standard deoxynitromycin. If it is replaced by the hydroxyl group, in case of compound 6, the activity dramatically fall to 400 micromolar. Compound 8 showed IC50 value 466 micromolar, and compound 9 showed activity 1200 micromolar. The kinetics profile of the isoflavone class of compounds are as follows. Compound 1 to 9 showed non-competitive type of inhibition, and among them, compound 1 and 5 showed strong binding affinity with PI values 63 and 31 micromolar, and where the IC50 values were respectively 114 and 40 micromolar. Flavonoid class of compounds, compound 16 to 20 showed IC50 values 50 to 499 micromolar, and flavonoid class of compounds 21 to 25 showed IC50 values 12 micromolar to 242 micromolar. We can see the same class of compounds has diversification in the biological activity, and the IC50 values varies from 1200 to 12 micromolar to 242 micromolar. Flavonoid glycosides showed IC50 value from 131.33 micromolar to 367 micromolar. Among all the flavonoidal glycosides, rutin showed the highest inhibition, that is 37.45 micromolar. And here we can see in the rutin, they are having disaccharide linkages. So this is the glycosidic part along with the flavonoid compounds. Kinetic profiles of the flavonoid class of compounds. Compound 16 to 19 showed linear mixed type of inhibition, other than all the compounds 20 to 26 showed 29 showed non-competitive type of inhibition. Here, compound 8 showed non-competitive type of inhibition, and compound 29 and 30 only showed these two compounds showing competitive type of inhibition, and compound 31 to 36 showed non-competitive type of inhibition. Deoxynitromycin had 425 micromolar. IC50 values and acarbose was 780 micromolar. So the drug present in the market showed quite high IC50 values, 780. On the other hand, natural class of compounds, many of the flavonoids showed very potent IC50 values. Here I'm going to explain the compound 30, the mechanistic studies, the mechanism of action as on the receptors, alpha-glucosidase. Compound 30 is a C-glycoside flavonoid, where R is glucose. Here we can see 1 over Vmax is a constant, so it is not varied. Here Vmax is not affected by increasing the concentration of compound 30 at 21.9 micromolar, 43.7 micromolar, 87.5 micromolar, and 175 micromolar. The apparent KM increases with the increase of the inhibitor concentration at the same sequence or order. Increase the KM cause decreased substrate affinity, which suggests that this compound competes with the substrate. The Ki is directly determined from the Dixon plot as well as from the secondary plot of line over breadth plot here in the right hand side of the diagram. The secondary Dixon plot also confirms the competitive type of inhibition as it crosses the origin, and the inhibitor and substrate binds to the basically in competitive type of inhibition. This is the basic biochemistry, and from the Michaelis-Menten equation, we know that the inhibitor and substrate, if they are binding for the same binding sites, so that they are competing for the same binding site. So that's why we know that the competitive inhibitors. And nowadays there are a lot of drugs which are actually non-competitive inhibitors as well. I was talking with one of my colleagues from Harvard University who is now working at Bayer Pharmaceutical Industries, and he's working on some receptor. So he is selling During our discussion, we find that we try to convince each other that non-competitive or even suicidal inhibitors sometimes preferred as the drug when there are crucial clinical conditions. The cytotoxic properties of the alpha-glucosid is evaluated on the cell lines, cervical carcinoma cell lines, and breast cancer cell lines. Here we can see a right-hand side, the cell growth is affected and they clumped, and the confluent cell growth, which is left-hand side, the control, is hampered. So the result was that most of the natural class of alpha-glucosid inhibitors were proved to be non-toxic in nature, except compound 6. The compound 6 showed IC50 values 400 micromolar. On the other hand, other flavonoid compounds showed greater than 100 micromolar. So we considered this, and from the literature review, it is confirmed that they are non-toxic in nature. There is no doubt natural class of compounds are more safer than the synthetic inhibitors or synthetic class of the alpha-glucosid inhibitors. The 3D QSR study of the flavonoid class of compounds. What is QSR? This is the most modern approach to rationalize the quantity structure activity relationship studies. QSR is a mathematical relationship between biological activity of a molecular system, understand its geometric and chemical characteristics. QSR attempts to find consistent relationship between biological activity and molecular properties. Thus, these rules can predict the activity of new compounds, and on the basis of the new molecules can be designed. And actually, there are new molecules that are designed on the basis of QSR, and I may walk you over there. So a 3D database is created on Civil 6.9 tripos associates, charges for each of the compound of 26 flavonoid characters calculated by gas tiger culture. Here, all the structures minimized using MMFF94 force field. And here, the ring A and B of the flavonoid compound were aligned by atom fit method. Here is only the sp2 hybridized carbon atoms were used. And due to its conformational rigidity, here, if we can see the first ring, this is in case of flavonoid, ring C. And the second ring is observed with the arrow, white arrow. This is the ring C in case of isoflavone class of compounds. Steric and electrostatic field energies were calculated in case of COMFA and COMSIA. COMFA is comparative molecular field analysis, and COMSIA is comparative molecular similarity indices analysis. The regression analysis performed by using partial least squares PLS methods. Here, is very interestingly, we had to remove actually 20% of the compound from the training set and reran the QSR and used it to predict the excluded compound. This was repeated for the 4 times. And finally, we get the R-square conventional correlation coefficient, 0.905 for COMFA and 0.935 in case of COMSIA. At the same time, we got the Q-score values, which is very important statistical significance, cross-validated correlation coefficient, 0.605 for COMFA and 0.602 for the COMSIA. And finally, we got the contour map. This is the contour map. We can see the different colors, regions. So those are the basically different regions of the molecules, which indicate which type of functional groups is preferred for further potent alpha-glucosid inhibitors. Here, in the background, I would like to show you that the blue-colored curve, here the actual log IC50 values of the experimented values in blue-colored column and the predicted IC50 value in purple-colored column for COMFA and COMSIA values are very close. For example, in case of compound 25, the actual value is 3.61. On the other hand, predicted COMFA value is 3.56 and predicted COMSIA value is 3.564. They are so close to each other, and that actually indicates a very successful models for these QSR studies. Here, the actual PIC50 values are on the x-axis, and the values are so close that we can see the purple and navy blue colors are superimposed with each other. Actually, they are superimposed, which indicates a very good models. This is the compound 29 for 20 COMFA models for compound 29 and 27. COMFA contour plot showed the statically favorable region. Green color is found on ring C in case of flavonoids. And excuse me. and sterically disfavorable region, which is yellow in color on ring C in case of isoflavone. Flavonoids are more active than the isoflavone class of the compound. Flavonoidal glycosides are less active as the glycoside residues are present sterically disfavorable region, which is yellow in color. In COMSIA model, here there is an additional sterically favorable region near C7 position, and the geranyl moiety of compound 5 is passed through this region. Thus, it can be suggested that bulky hindered group at C7 position may further improve the activity. Perhaps it has hydrophobic interaction with the enzyme binding site. And I would like to mention that here there is a research group who reported in 2024 that they synthesized some molecules and they added the bulky glycosidics and other bulky functional groups at the 7th position on the geranyl moiety, and they find with the similar kind of IC50 values against alpha-glucosidase. This compound was had IC50 values 40.73 micromolar, and which was 10.4 times more active than the standard drugs. Besides this, we can I cannot discuss the details, but I can show some new classes of natural alpha-glucosidase inhibitors. This is the acyl fluoroglycinol isolated from Metis communis, and they are also showing very interesting IC50 values with 46. Compound 2 showed 46, 46 micromolar, and compound 1 showed 84.3 micromolar concentration. And tritropin is the most exciting class of alpha-glucosidase inhibitors in our studies. And this is a novel tritropin alpha-glucosidase inhibitor we reported back in 2008. So here, deoxynitromycin was the standard. This was 4.25 times more active. This third ring is a 7-member, which gave it its new novelty as a tritropin class of compound. So the compliant was Duboisia marcopa. Dubulan is a novel tritropin skeleton which showed IC50 100 micromolar, which is 4 times more active than the standard. A total 31 tritropin from different classes were included in the study. Some of them presented in this discussion, but hopefully next time I will show those. The iridoid was a new class of compounds, and it has fluorimycin showed IC50 value 135 micromolar, and isofluorimycin shows the similar kind of alpha-glucosidase inhibition. It is isolated from plumeria rubra, and these are the flumyricin. So isofluorimycin was 3.2 times more active than the standard. On the other hand, flumyricin, they are geometrically isomer to each other, as you can see from the structures, but they showed almost similar effect on the alpha-glucosidase inhibition. Their mode of inhibition is also the same. Both showed non-competitive type of inhibition, which indicated that the binding affinity of the compound perhaps is not dependent on these peripheral C14 methyl groups. Pisolin was new class of compounds. Unsaturation at C5 and C6 is very important. IC50 values 187.5 micromolar. If the unsaturation is converted to epoxide ring, the activities drastically fall to 782. When there is a hydroxylation at C5 and C6, the activity further decreased. Antranol is another interesting new class of compounds we reported. This is from Haraguana madagascaris. When the prenyl group is replaced by the furan ring, the activity is decreased 21 micromolar. So here, the prenyl group definitely is playing a crucial role for binding with the active site of alpha-glucosidase enzymes. Acridone is another new class of inhibitors. Among all the acidones, phenylated one, which is compound 3, showed highest activity with 17 micromolar, which is also involved in the hydrophobic interaction with the enzyme. And compound 2 is a dimer, which is also showing its steric effects in the inhibition of alpha-glucosidase. Finally, we got the homology model for alpha-glucosidase. As I already mentioned, that there is no crystal structure of alpha-glucosidase 3 to 120. And this is one of the compounds, compound 32. And we can see the docking studies showed the phenyl 291 and phenyl 148 and phenyl 302, which are playing crucial role in the binding with the natural compound. Reserpine type indole alkaloids. Here, compound 1 showed IC50 value 75 micromolar, which is further increased to 55.34 micromolar in case of compound 2. Conjugation on ring D seems responsible for the enhanced activity. Absence of the unsaturation and ketonic functionality on ring D. In case of compound 3, activity is decreased to 800 micromolar. Perhaps the flexible ring system hindered binding with the enzyme and decreased the activity. Discoumaring, another new class of alpha-glucoside inhibitors. If this in this R here this R group is position is nine membered aliphatic chain compound 2. The IC50 value is 4.4, which showed very high activity. And if it is substituted by 7-membered carbon chain, the activity increases 60 times than the standard. If there is only hydrogen, the activity is not at all satisfactory with high IC50 value, 1200 micromolar, which dictated that the aliphatic chain with 7 to 9 member increases the activity, as we found in same in case of flavonoid compound, zeranol moiety is responsible for increased activity. So these are all. Thank you very much. I would like to acknowledge to us the World Academy of Sciences, Trieste, Italy, for hosting my PhD at the International Center for Chemical and Biological Sciences, and my sincere gratitude to my professor, mentor, and supervisor, Professor Dr. Muhammad Iqbal Chaudhry, and my co-supervisor, Professor Dr. Ataur Rahman, along with a lot of foreign faculty members who taught me at HEJ Research Institute of Chemistry. Thank you very much.
Thank you very much, Dr. Shamson, for that very comprehensive presentation. Any questions from the audience? Anybody? Okay, tell me a little about the anti-glycation property of the compounds that you looked at.
I think, Dr. I'm waiting for that to do the anti-glycation work with you. So I think next time. But alpha-glucosidase inhibitors prevent glycation as well, because for the glycation process, alpha-glucosidase enzyme is important for the glycosidic linkages, and inhibits the linkages. So that's why it is very important to have the anti-glycation properties as well. And actually, I'm looking forward to work with Dr. So Dr. Samba here, the chairing the session. So detailed mechanistic study we will be able to do with him, inshallah, next time.
Yes. I think there's a question on the line as well for you.
So, so we have more than 90 participants online. So I'll read the questions that have been posted for you. The first question is, can we use the same method for anti-diabetic peptides or protein?
Yes, definitely. One of my work was with the TAF peptide and 7sp small nuclear RNA interaction. And isothermal calorimetric assay is the wonderful assay I have done at Harvard University Molecular and Cellular Biology Department. The difficulties is the machine was very expensive, and when I was working with these machines, so yes, definitely we can work with the short peptides, and because there is protein-protein interaction now, it's pretty much established. When we were at the beginning of these studies, the protein-protein interaction was, especially the NMR studies on protein-protein, was not that progressed that time era. Now it is very much progressive, and even we can use NMR to see the structure, because I had the training in structural biology as well. So we can see but the condition is not more than 100 amino acids of the peptide. So short peptide would be very great. And for the structural biological part, we can use 8 to 9 or I think 10, 15 peptides even. These short peptides are easy to synthesize in the laboratory, and we can do it. And it's obviously it's possible. And I think we should move forward to the more biological studies, because the next drug discovery and design will be basically on peptides, RNA, and DNA. Thank you.
So thank you very much, Dr. Shams. I hope that the participant who posted the question has received the answer. So let me read some other question that has been posted for you. Okay, so this is from Miss Mariam. She's asking, what is the cause of flatulence associated with acrobos, and will these natural products prevent these side effects? Because I believe that is a major prompt to looking for these natural product inhibitors.
Thank you very much, and these are wonderful questions. And just recently, I addressed this question when I was editing one of my book to be published by Taylor and Francis publishers. Actually, natural molecules and its architect is very compatible with the biological receptors, and this acarbose is not a natural product. Rather, it's a synthetic derivative. So this is the amino sugars like structures. So definitely there is always complications regarding the synthetic drugs, and there is always positive effects due to the compatibility, because our biological systems, our bodies, these natural receptors and the natural compounds have some sorts of compatibility. That's why thousands of years Ayurveda and different Unani systems are sustained in the subcontinent. We should not forget that, and we should conserve the medicinal plants and should be conservative really regarding the natural products. Thank you very much.
Okay, so I think there is one thing leads to another. So there is another question. Can we use QSAR for biopolymers?
Thank you very much. This is a very interesting question. So I need to look into it. For the 3D QSR, really, we need to have molecules which has comparatively static structures. That means we need to have sp2 hybridized carbon atom for a very productive 3D QSR study. That was the limitations for vanadium complexes when I was studying vanadium complexes and triterpenes, because as you can see, the triterpenes, there are hardly a few sp2 hybridized carbon, which is not enough to have the 3D QSR studies. So same things will be applied for the peptides and biomolecules, because there are very few sp2 hybridized carbon atoms over there. So every method has limitations, but hopefully in near future there will be some softwares, as this is a different world, different era, and there are a lot of computer sciences program developing. Thank you very much.
Okay, thank you very much, Dr. Shamsun, for that lovely presentation. Right, I'm going to introduce the next speaker, who is Mohammed Ashraful Zaman. He is a senior research fellow at ANRAP, University of Health Sciences, Dhaka, Bangladesh. His research interests are diabetes, cancer, natural products, RNA modification, and its cellular feedback. He was awarded the Guangdong Government Outstanding Foreign Student Scholarship in China. He was also awarded comstech Research Fellowship for Least Developed Countries, again in Islamabad, Pakistan. He is a registered pharmacist who earned a Bachelor of Pharmacy degree, followed by a master's degree in biotechnology from Bangladesh. And he has a Master of Philosophy degree from Hong Kong. He's been actively involved in research and university teaching for the past 8 years. And during this time, he has received several national and international fellowships and scholarships. As part of his research accomplishments, he has published 7 articles in peer-reviewed international journals with high impact factors and 1 book chapter. Over 150 citations are listed across various research profiles. He's going to talk about anti-liver cancer effects and molecular mechanisms of flower extract from Camellia nitidissima, CW chai, based on network pharmacology and in vitro cell culture validation. So over to Mr. Ashraful Zaman.
Thank you, sir. Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullah. At first, I'd like to convey my gratitude and venerations for our chair, my respected teacher, my distinguished scientist, my brother and sister in front of me, and definitely the organizer. This is Mohammed Ashraful Zaman, and the next few minutes I will talk about this topic according to this content. The first, I will discuss about the research background and then go for the network pharmacology work. And in chapter 3, I will discuss about some basic chemical study that I did. And the finally, the in-depth biological study. So for hepatocellular carcinoma is one of the main type of adult primary liver cancer, and it is the third leading causes of death globally. According to the WHO report, more than 1 million people will die from liver cancer in 2030. More than 70% of all HCC cases occur in Asia. China comprises the majority of cases in Asia, followed by Japan, India, Thailand, and Vietnam. So researchers are still working on alternative therapy for cancer because the limitations of existing therapy. So for example, liver cancer have only two kinds of treatment. One is surgical treatment, and second is the non-surgical treatment. And arterial liver chemotherapy, chemoembolizations, ethanol injections, radiations, and systemic therapy belongs to the non-surgical treatment. However, several limitations of this treatment still exist, support the viral infections, lower survival rate, lower success rate, poor understanding of molecular mechanisms. Some fundamental limitations are multi-drug resistance, high cost, long treatment durations, treatment facilities are not available, and finally, severe side effects exist. So Camellia nutritissima is a medicinal plant. It has been used traditionally as tea and also against several diseases for long. Wild C. nutritissima is distributed particularly in the Guangxi area of Southeast China and also cultivated in different provinces in China. And interestingly, this plant also included in the Chinese encyclopedia called Ben Kao Gan Mu. So the rich chemical properties of this plant are responsible for several biological activities, including anti-cancer, antioxidant, hypoglycemic, hypolipidemic, anti-allergy. And the recent scientific evidence showed that this flower regulates molecular signaling pathway, including PIK, AKT, MAPK, PI3, AKT, MTR, and gives potential effect against cancer. The rationale of the study said that the 2.9 billion carcinoma deaths and 4.3 million new diagnoses annually will the urgent need for innovative HCC treatment among Chinese researchers. China's rich traditional medicine heritage offers a unique opportunity to develop accessible novel anti-cancer drug by integrating ancient practices with modern pharmaceutical approaches. C. nutritissima, a medicinally valuable plant. If the flower of nutritissima are harnessed as a raw material for anti-cancer drug formulation, it could lead a significant health benefit for the purpose, particularly in combating HCC. The successful development of an anti-cancer drug would not only improve public health outcomes, but also provide substantial economic advantages. It boosts the world's healthcare system and enhances its standing in the global pharmaceutical market. Additionally, it aligns with the international health policies aimed at reducing cancer-related mortality. So the research gap has said that different researchers have predominantly focused on cancer other than the hepatocellular carcinoma, notably the MAPK ERK pathway, critical for HCC treatment has not yet been explored in relation to C. nutritissima flower. There is current no evidence from in vitro or in vivo studies demonstrating the impact of C. nutritissima flower extract on vital signaling pathway. Addressing this gap is crucial for understanding the potential of C. nutritissima flower as a therapeutic agent for HCC. So the aim of this research was elucidations of a potential mechanistic target for anti-HCC therapies. It belongs, three objectives are belonging under this theme. And the first one is to investigate the potential therapeutic anti-age, which is the target for network pharmacology, to investigate the chemical properties, and finally, to evaluate and to validate the therapeutic anti-HCC target using signature stemma flower active extract and explore its mechanisms in vitro study. So this is the research design. At first, we collected the dry flour and then go for the grinding process and extraction process. After completing the extractions, we use the in vitro study using two kinds of cell line, HCC-LM3 and HEPG2. Simultaneously, we also performed the chemical analysis focusing on the TPC, TFC, and some antioxidant properties. Therefore, after the primary evaluations, we select the bioactive crude extracts as well as the crude extract sensitive cell line. After selecting the bioactive crude extracts, we go for the chemical identifications by LCMS, and then go for the network pharmacology and predict the signaling pathway. After predicting the signaling pathway, we again repeat the in vitro study and focusing the RT-PCR, Western blot, cell cycle, and apoptosis necrosis scenario. Finally, we recorded our result. So this is the opinion. This is the chapter 2, and describing the network pharmacology, which was first proposed in 2007, offer an efficient and systemic approach to elucidating the molecular and pharmacological mechanism underlying the traditional medicine. The aim of this chapter is to achieve the objective number 1, the primary objective number 1, which is to investigate the potential therapeutic anti-liver cancer target through network pharmacology. It belongs to intermediate objectives. The first one is find out the target genes for bioactive compound. Second one is the screening gene responsible for the liver cancer. And finally, to predict the molecular signaling pathway and drug protein interactions by enrichment analysis and molecular docking. So this is the methodology that I performed, and this is the result. At first, we developed two state of gene bank. Which one? First one is the liver disease-related gene bank, and second one is for the compound-related gene bank. And then we use the Venn tool to combine them and try to collect the common genes from these two kinds of gene bank. And after collecting the common gene, we use the cytoscape to make a PII, protein-protein interaction network. And from here, we identify the most top 20 proteins which have the same rank. And these 20 genes was used for the ShinyGo online tools to get the top 60 signaling pathway. And here we select, we find that the 3 signaling pathway that we are interested against our treatment, this apoptosis signaling pathway, the AKT-P1-3 signaling pathway, and the last one is the MAPK signaling pathway. This is the interactions of different signaling pathway. So our target is the apoptosis signaling pathway, PI3K AKT signaling pathway, and the MAPK signaling pathway. From this figure, we see that so there are different relations of with the other signaling pathway. So therefore, we constructed a compound target pathway network using the cytoscape. So here we found that the 17 compounds which is identified by the LCMS, we will discuss later the next chapter. These 16 compounds are actively influencing these 10 genes. That 10 genes are regulated these 3 signaling pathway, which could be beneficial for the cancer treatment, liver cancer treatment. So therefore, we select this 3 signaling pathway and 2 genes, BCL2 and AKT1 for our interest. And therefore, we found that the BCL2 protein are regulated by this 8 compound, and the AKT1 protein can be regulated by this 8 compound. Therefore, we go for the molecular docking, and we found that the procyanidin with C1 has a maximum binding affinity with the AKT1 protein, and the neuriocytrin has the maximum binding affinity with the BCL2. So from these observations, we can assume that these 2 kind of compound may be a good choice for modulating our target proteins as well as our signaling pathway. So this is the end of this chapter 2. And now the conclusion part is the current investigation revealed that 16 compounds of the C-nitrosamine flower extract can regulate 4.5% of genes that are closely associated with the liver cancer progression, whereas the MAPK, PI3K, AKT, and apoptosis signaling pathway could be a good regulators. The compound procyanidin C1 and neuriocytrin have high chances to regulate BCL2 and AKT1 protein. So for the next chapter, the chemical study, the aim of the third chapter was to achieve the main objectives too, that is to investigate chemical properties of chemical C-nitrosamine. And the 2 intermediate objectives is also there. And the first one is the biosegregated extraction using the 4 solvents. And the second one is to find out the best extract based on these parameters. And finally, to identify the chemical compound of the best extract using the LC-MS. So this is the methodology, the basic methodology of my work, and this is the extraction summary. So for the result of total phenolic content and total flavonoid content, we found that the ethanol extract shows the maximum quantitative. And for the antioxidant scavenging activity, we found that the ethanol extract also show the maximum in FRAP techniques. So from this basic data and as well as some cell line data, we selected the ethanol extract for the LC-MS analysis that I said in the last chapter. And this 25 compound, we found more than 2,000 compounds in by the LC-MS analysis. And from this 2,000 compounds, we select top 25 compounds based on the abundance score. And this 25 compound has been isolated. And finally, the ethanol extract of C-nitrosamine contains a remarkable amount of TPC and TFC. Ethanol extract contains high FARP free radical scavenging capacity. The 25 most significant compounds are identified by LC-MS. This is the end of the chapter 3. Now I'm going to deliver the chapter 4. This study mainly used 2 experimental models in vitro liver cancer cell research, the HEP-G2 and LHCC LM3 cell line, to perform a deep investigation and analyze the anti-HCC effect of C-nitrosamine. The aim of this chapter is to achieve the final objectives, that is to validate the therapeutic anti-liver cancer target using C-nitrosamine flower extract. And it also has 2 intermediate objectives. The first one is screening the potent extract and cell line based on LD50. The second one is to investigate the anti-cancer effect of C-nitrosamine. And finally, to elucidate the signaling pathway associated with the anti-cancer effect. This is the methodology that I used to validate the biological activity. The first one, I used these 2 cell lines with different treatment durations, 24, 48, and 72 hours, with different doses to 125 to 10 microgram per ml. And AI is, use the CCK-HSA to identify the LD50 and select the doses. After selecting the doses, I use the colony formation test with the both cell lines and try to find out the best cell line. And after selecting the doses and best cell line, I used the flow cytometry and the fluorescence microscopy for identifying the apoptosis and necrosis. After confirming the apoptosis and necrosis scenario, I go for the Western blot and qPCR to investigate in deep molecular mechanisms. And thereafter, and then I found that in the cell viability test, the N-hexane extract affect the HCC-LM cell lines and with a near about the 80%, which does show any dose-dependent effect. And for the HEP2, there is no changes of cell viability. For the DCM extract, we found that it shows the cell viability about the 40% with the dose-dependent effect, and also for the time-dependent effect. The HEP2 cell line also shows that same as like the HCC-LM cell line. For the ethanol extract, we found the cell viability was only 15%, which shows the dose-dependent and time-dependent effect. For the HEP2 cell line, we also found the about the 15% cell viability showed the dose-dependent effect. For the water extract, we found the HCC cell line shows the 85% cell viability with no dose-dependent effect, and about the 90% cell viability for HEP2 cell line with no dose-dependent effect. So we also use the cisplatin as a standard drug, and we found that the cisplatin shows for 30% cell viability for HCC-LM cell line and for the 50% cell viability for the HEP2 cell line. Therefore, we assessed the LD50 and found that the cisplatin shows that 50 to 44 to 55 microgram per ml for both cell line, and ethanol extract shows the 340 to 425 microgram per ml. So in interest, and other extract didn't show any results. Maybe it needs more high doses for getting this value. So this is the fluorescence microscopy scenario. It's very clearly seen that the ethanol extract shows the dose-dependent cell viability. And if the dose is high, the cell viability is low. And this is the HEP2 cell line scenario. This two scenario pattern is same and supported the cell viability results that I showed before. And therefore, we go for the cell line selections, and I use the colony formations. So for HEP2 HCC-LM3 cell line, no colony formation was observed after ethanol extract treatment. And for the HEP2 cell line, undermost colonies at the lowest doses were observed after the ethanol extract treatment. So therefore, the ethanol extract with the dose of 250, 50, and 550 microgram per ml has been chosen for the further in-depth biological study, focusing on the necrosis and apoptosis scenario on HCC-LM3 cell line. So for the necrosis and apoptosis, red spot represents the necrotic cell, and green spot represents the apoptotic cell. From the fluorescence microscopic picture, we found that there is plenty number of the red spot, but there is no green spot. But when we analyze this picture by image software, we found the mixed model of cell death. So the necrosis and apoptosis both are present there. So for confirming the apoptosis scenario, we go for the flow cytometry. And we found that the fluorescence intensity of GO and G1 phases was not significantly higher compared to the G2 and M phase after treatment. So extractive treatment leads a higher number of apoptotic cell populations at G1 phase compared to the control and versus treatment. Therefore, we investigated the cell signaling, cellular signaling, focusing on the ERK1/2 using the Western blot. We found that TNF-alpha dose dependently expressed phospho-EMK by MEK1/2 ratio also increased. However, the phospho-ERK ratio significantly reduced. So we arise the questions why it's happened, because their signaling system cascade as the same. So therefore, we select more genes for identifying this one. We found that the DAUSP5 significantly increases dose dependently. DUSP1 and HRAS also expressed compared to the control. So therefore, we predict a model of linear model of this mechanism. We found that we created that if the TNF-alpha is stimulated, therefore it influences the HRAS, MEK, and then ERK1/2 in the cytoplasm. After activating ERK in the cytoplasm, it goes to the nucleus, and after entering the nucleus, the DSP overexpression inhibited its expression. So there is one of the limitations of current study is the expression level of ERK in the cytoplasm and expression level of ERK1/2 in the nucleus is not identified. So this is one of the limitations of this study. And therefore, we go for another signaling pathway that is cascade signaling pathway. Protein expression was observed by Western blot. We see the bugs and cascade signaling proteins decreased dose dependently, whereas the BCL2 interferon protein is increased. And interestingly, the cascade-3 is also significantly increased. So therefore, we expanded our molecular model pathway, and we found that this cascade-3 may be important for this apoptosis scenario. However, we didn't perform here the active cascade-8, so we can't say that this signaling pathway, apoptosis signaling pathway, are responsible for the apoptosis. Therefore, we go for another investigations for the phospho-PI3K and AKT ratio. And then we found that the phospho-PI3K and PI3K ratio reduced dose dependently. The phospho-AKT also reduced significantly. And therefore, we could expand our model that this, if once that TNF-alpha expressed, it influences the HRASs, and HRAS also dephosphorylated the PI3K as well as the AKT, and stopped the cell survival and growth factor. So this is the total model of my investigation. However, we have several questions about this one because we still not confirm what is the scenario of mechanistic scenario of necrosis, because we don't yet did some work like the current work study did not observe the modulation of transmembrane depth receptor like CD95 trial MLKL active cascade-8. Therefore, this study does not provide any causal link between the apoptosis and necroptosis pathway. In conclusion, ethanol extract showed time and dose-dependent necrotic effect on the H6LM cell line in the microscopic observations. Therefore, the gene expression of USA5 and USA1 was confirmed, whereas the TNF alpha mediated activation of HRAS, HRAP, MEK 1/2, ERK 1/2 signaling pathway may play a regulatory effect on the cell death scenario. Activation of ERK 1/2 may lead the expression of the cell 2 and dephosphorylation right of caspase-9 and Bach's protein. Caspase-3 may have the regulatory effect on the cell death scenario. Once HRAS expressed, PI3K AKT protein may be phosphorylated. So this is the references that I used in my presentations. I must acknowledge the Professor Shu, Professor Sai, Professor Shukza, Professor Begum Rukia, Professor Peter, Professor Andrew. I acknowledge all of the members of the Faculty of Life Sciences, UIC, Hong Kong Baptist University. I acknowledge the Department of Education, Guangdong Province, China. I must state my heartiest acknowledgement to ANRAB, BHS, and all of my colleagues for nurturing my research career. It is my pleasure to show a deep acknowledgement to the ANRAB OIC comstech joint approaches to arrange such a beautiful regional seminar. Finally, I acknowledge my beloved son and daughter, sweetheart wife, my respected parents, and all siblings for their support, love, and encouragement. Thank you. Thank you so much.
Thank you for that wonderful presentation. You've obviously done a lot of very, very good work. Over to the audience. Any questions? Anybody? Okay, there's a question there.
Thanks for comprehensive presentation. I have a question related you have used the ethanol extract having potent activity against that HepG2 cancer cell line. So what could be the reason that the DCM extract, the hexane and other extracts, they have lower activities as compared to the ethanol extract? Second one, I didn't see the ethyl acetate extract. Like in the LCMS analysis, most of the compounds which have been detected, they are flavonoids. So ethyl acetate is a suitable solvent for the extraction of flavonoids. Thank you.
Thank you, sir, for your question. Actually, at first I used the post-segment based on the polarity. So I'm trying to identify, separate the compound according to the polarity. So I just randomly choose the solvent. And the second question is, I didn't use the ethyl acetate solvent, right? So my question is that this flour is normally used as a drinking tea. So that drinking tea is the water soluble, right? So that's why I might focus was to polar solvent, which can normally we can find out by using the ethanol extract. Yeah.
Any further questions from the audience? Yeah, there are some online questions.
So the first question, did you use also DPPH?
Yes, I also use that DPPH, and the result of this one I showed in the slide also.
Okay, okay. On which basis you choose BCL2 and any other gene in the network pharmacology?
Actually, there is no method for choosing this one. We just choose according to our lab facilities and according to our intentions, because our intention is the apoptosis, and BCL2 is one of the apoptotic proteins. So I choose the BCL2.
So there is another question. First, praises for your excellent work. what is the safety profile of the flower extract?
Actually, it's a natural product, and in China, it's consumed as a tea regularly. So I didn't did any performance for the identifying the safety profile of that flower.
Thank you so much for the nice presentation. For the extract, many times you haven't taken the extract for your use in the research. Have you standardized the extract, or you have any methodology that every time that you use the extract has been standardized and have the same or approximate same constituent?
Sorry, sir. Would you speak again?
The extract you used,
Yeah.
have you standardized in any way that you can say that every time or in next future, when we'll use the same extract, will give the same results or like this? Do you have any methodology to standardize the extract?
No, sir. I didn't make it standardized, but I just follow the established protocols to do the extraction. And I believe that if anyone can do the same procedure, they can get the same result.
But it is a suggestion.
Yes, sir.
That-
Yes, sir.
Every time you need to, you need to standardize, because every time extraction, heat, time, and many things influence the phytochemicals. And if you standardize it scientifically, rationally, then you can focus. Thanks so much.
Thank you. Thank you.
One final question? No? Okay. I think the question goes like this: why we use flower? Have we used other parts of the camellia nitidissima plant? Why use flower?
Actually, a flower using for the natural drink. And so that's why I choose the flower.
Okay. Okay. One final question: are there any preclinical in vivo studies for the extracts, or have you got something planned for the future?
For the future, not yet, but I'm planning.
Okay, okay, thank you. I think that's it. I think that concludes this session 3. So thank you to the 3 speakers for their wonderful presentations. Thank you to all of you for being such a wonderful audience, okay, for engaging in, the question and answer activity. So thank you very much. I think we have a tea break now. Is that right?
Yes, we have tea break, and we will convene our next technical session here after 20 minutes of tea break. Thank you very much. Okay, Bismillahirrahmanirrahim. Welcome back, ladies and gentlemen. So we're beginning with the 4th technical session of our 2-day regional seminar of 8th ANARAB and COMSTEC seminar. So this technical session will be chaired by distinguished national professor, Professor Dr. Abdul Kalam Azad. He's the president of Diabetic Association of Bangladesh and chair of Center of Global Health Research as well, Health Research Diabetic Association of Bangladesh, Dhaka. So I request you all to please welcome our chair. Sir, I hand over to you to introduce and welcome our speaker.
I've already introduced. And she would be presenting on enabling technology in drug discovery.
Assalamu alaikum. Thank you so much, sir, for your kind introduction. So today my talk actually is divided into two parts. One is specifically for students. I'll be discussing about the new technologies that actually enhance the drug discovery process. And then I will be discussing about the anti-glycation work that our group has done. And for that, we were taking constant advice from Dr. Nisar Ahmed. And as we understand, the drug discovery process, it's about 10 to 15-year process. And so it divided different phases. For example, which is called pre-discovery phase, that most of researchers are actually working in this area, which is called drug discovery. We are identifying several molecules from natural sources, from synthetic molecules, and these various type of the molecules from combinatorial libraries, etc. And then we evaluate these molecules against different targets. That targets could be a protein, enzymes that can bind to it or regulate their work. Specifically, there are many proteins that are relevant to enzymes overexpression. So we usually target the inhibition of that proteins or enzymes. And then we also do preclinical studies quite often. We establish different in vivo models after in vitro studies, and then evaluate these molecules against in vivo studies. But the phase that we always have some problem, I think, as a researcher, that's my personal opinion, that we identified many hit molecules, but we cannot take it further. So here the pharmaceutical company is playing a very important role, but this require different clinical trials, like phase 1, phase 2, and phase 3 clinical trials. And for these clinical trials, for the phase 1 is usually efficacy studies and safety studies that has been done. And phase 2, actually safety studies in phase 1 and efficacy studies in phase 2 usually. And the number of volunteers are increasing. So in phase 1, we have healthy volunteers, and phase 2 are for patient studies on patient, and on the large scale, the phase 3 clinical trial. And then it goes next step for the drug that could be registered. And there are many drugs that are fail in phase 2 and phase 3 clinical trials. And we also know that even after marketing, few years of marketing, the companies actually withdrawn these drugs from market because of the side effects that been observed over the period of time. And this you could see number of years, that about usually 10 to 15 years time are required. And if you could see the number of compounds that we could identify in the initial phases, like 10,000 to 15,000 compounds, and then for the preclinical, we can take up to 250 from these data, and like 5 compounds goes to the clinical trials, and maybe one FDA-approved drug if some company or someone is so lucky to have a drug in his or her lifetime being approved from the Drug Regulatory Authority. And the overall cost is about $1 billion US. So, and being the technologies that the new technologies that has been developed over the years. So actually, the drug cost of the drug development process is being decreased from 2.8 billion US to 1 to 1.3 billion US. And it's not only about the money, it's also the time factor. But we have seen the historical milestone during the COVID pandemic time, and in which the new vaccine has been developed and come to the market in a very shortest period of time in the human history. And not only these, drug repurposing has been also played a very important role. There are many drugs which are already in the market has been identified to use for the treatment of COVID-19 patient. And therefore, the whole process that has been moving forward and the new technologies that has been developed in last few years are now playing a very important role. And as a scientist, we also need to understand that how we could and learn how could we relate our research with these, and we can use these technologies to make our researches to move further and try to get benefit on these. So, for example, artificial intelligence, and which actually plays a very important role, and is to accelerate the drug development process. It's from identification of the right candidate and identifying the right compound, and to improve the manufacturing process. The overall is playing an important role. That next technology that are very important, it's big data. And that actually we as a researcher can use these big data for the small chemical compounds to genome or proteomic data to analyze the interaction of these compounds and to understand and get the information from these big data which are available. And then the quantum computing system that actually, and I think researchers are quite, now it's very frequent that we are using different type of cluster computers and quantum computing system to model the molecular structure and how the interaction of our hit molecules are interacting with these targets. And then gene editing, which is very important, and it's a new approach actually, and in which the target, the gene or tablet gene, when it is inserted in that specific location in the genome, and that can be considered for the therapeutic purpose, and repositioning or repurposing of the existing drug that we have seen now during the COVID time, a very enhanced level of repositioning and repurposing of the existing drug to discover the new use of these molecules. So with this, the second part of my presentation, which is more like a trivial, and I presented you here specifically for a student that we need to correlate or try to use these technologies and incorporate in our research if you would like to survive further next 20 years of research, because science is really moving further. And as Dr. Iqbal Chaudhry is my teacher, so he always says to us that in science there is no plateau. If you learn, then you move up. And if you do not learn and stop learning, you will go down. So there's no plateau. So we always, as a student, as researcher of science, we always strive to learn new technologies, whatever are developing. So the next part of my presentation is relevant to the workshop title, this symposium title, and we have identified the potential of some medicinal plant for their anti-glycation activities. And we were discussing these over the period of time with Professor Nisar Ahmed during his visits and also during online meetings. So we have screened about 111 plants and in vitro studies, anti-glycation studies, and out of them, 22 were found to be moderately to the potent activity. And then we have done the in vivo studies, as I showed you in the first slide, that the preclinical studies that scientists can easily do. The in vivo studies first for toxicity and then S2C-induced diabetic rat model. And then we also checked the LD50 during the toxicity. And then we have done some further biochemical work. So, and the reason behind it, though we have several single molecules that are present in the market, but still the world is going towards nature. And if we could find out, and I'm not going in details about the diabetes and because we are listening all these things for since yesterday. So we understand the whole cascade of diabetes and the problem that involved to it. But we would like to identify some plant from nature that can help to relate this diabetic related complications and reduce the glycation effect. So the in vitro studies that we have done on these are the results that you could see. There are different medicinal plants, then their name of the plant and common name, and their IC percentage inhibition and IC50 values. So and here there are some. I'm presenting here those only which give a good anti-glycation potential. So you could see the value from these are in micromolar 0.307 micromolar, 0.7 micromolar, and 1.4, and as compared to our standard. So then the selected medicinal plants we have checked for the acute toxicity, and during the acute toxicity we have given 24-hour time to see the results and try to find out the LD50 value on different doses. So we have the control group, the vehicle group, and those which are given the plant extract. And there are different doses that we have given, like 200 milligram per kg body weight and 400 milligram and 816 100 milligram per kg body weight. And the animals were observed continuously and monitored for 24 hours for any sign of toxicity and lethality. And those animals which have survived, they were anesthetized and dissected. And then the different toxicity parameters in blood have been measured. So here, these are the results of LD50, and you could see one of the plant is showing about 25% at 1600 milligram per kg body weight toxicity, but most of them, and there are these two plants which are showing 50% mortality rate with very high dose, which is 1600 milligram per kg body weight, and all others are safe at this study. And then we have done the streptozotocin-induced diabetic studies on these models, and we have selected among from this short listing, we have selected these medicine plants, and that we have done for in vivo studies on STZ-induced diabetic rat. And so in this model, we have again the vehicle control group and the positive control, which as a positive control, we have metformin treated group and the diabetic control only STZ group, and then we have group with which we have treated with 400 and 800 milligram. There are two different groups which were treated for this. And then after the treatment of about 1 month, and it was dissected. So here are the studies on these medicine plant that you could see that the blood glucose level, which was in control, and metformin in control rats, which was very low, and in metformin is also quite good, that the diabetic rat is showing very high level of blood sugar level. And with this these plants, you could see specifically Polygonum bristota, which is reducing the blood sugar level. And these are, the orange one are with the treatment with 800 microgram per ml, and the blue one is 400 microgram per ml. So, and here is the creatinine level that was again measured, and we found that Polygonum bristota and Circus infectoria is also playing an important role as compared to diabetic model. And the blood urea level and direct bilirubin level. So these two plants were found to be a very good impact on the blood, the important markers of the diabetes. And here is the standard value that we have taken and compared. And then the next step is we have checked the HbA1c level, and we could again find out the HbA1c level with the Polygonum is it's somewhat better than as compared to the other. And then we also checked the renal HEE and in kidney, and then it was also find out that these Polygonum bristota and L-menthol and Circus infectoria gave us quite good value. And we also estimated the hydroxyproline and total collagen content from the tail. And then we have find out these are the results of these. And here you could see that as compared to the control and metformin, these values are, which is quite relevant to the control, and control means non-diabetic rats, and the total collagen level and hydroxyproline. And so here, these are, we have selected some plants. So we have selected some medicinal plants that we checked for their anti-glycation activity on, there are a few were found toxic, and with 1600 milligram per kg body weight while there are the Polygonum bristota, Curcus infectoria, aloe vera, and L-menthol shows the good hyperglycemic and renoprotective effect. So these a medicine plant overall shows a very good anti-glycation activity, and it can be further taken further for maybe larger scale preclinical studies we can do, or maybe phase 1 clinical trial, but definitely we need work on this. A very short glimpse that I'll also show was one of the small part of our work that we do on alpha glucosidase. Again, it's an important enzyme, and Dr. Shamsunahar has presented very well. So I'm not going through the detail of how it functions. The important thing that you would like to mention here, that we also, since it's a pure enzyme, so we not only the in vitro activity bioassay that we have done, but we also checked using NMR spectroscopy, saturation transfer difference NMR spectroscopy, and these were the standards that we have used, the ACARPOS and 1-deoxynitromycin, which is one is the clinical standard and other is the research standard, and we have found quite good values and definitely kinetic value, and the STD-NMR that we have done to identify and the which part of the ligand is binding with protein, which is called epitope packing. So and not only with the standard and other compounds that are active, we also done STD-NMR with ligand which did not show any activity in bioassay. So we also found that there was no STD signal was observed. So which also an indication that, but definitely NMR is not a high throughput technique as compared to the any fluorescence or bioassay method. But we can check it to find out the epitope pattern. And there are different classes that is being checked. And we could definitely find a good IC50 value as compared to the standards. And I would like to actually show the STD-NMR effects. So here, this is one of the molecules that shows interaction. This is active and also is a competitive inhibitor. Using kinetic, we have done this and we identify, okay, this is the structure of the molecule. So these are the protons. So this part of the ligand is actually binding with the protein. And this, with the potent compound, which were active, we have done these binding study. Again, another molecule of the same class that shows the interaction of this part of the ligand, which binds to the protein. And here, so this is something different. So I'm not going in much more detail of these almost similar type of the results that we could get, that Dr. Shamsunahar also discussed. So various class of compounds that's been identified as a alpha glucosidase inhibitor and their mechanism of action through kinetic study was determined, and also the STD-NMR, which was the new part, is being included here. And an NMR is actually when STD is a technique in which a protein binds to the ligand, and you saturate the protein. You give a pulse to the protein, and then saturation is transferred through spin diffusion to the bound ligand. And but there is a limitation in STD-NMR. If the ligand is very tightly bound, then you cannot see the difference NMR spectrum. And here again, if the ligand is very loosely bound, then again we cannot see the difference NMR. But there are some values that that's a limitation, that it should be a reversible reaction. Means the ligand has to come and bind with protein and then go back. And we use very high concentration of the ligand in protein in FTP-NMR as compared to the protein, about 100 and 200 folds more. So I would like to acknowledge my students, Salim Halalani for alpha-based work and anti-glycation work. Dr. Sabah Taha and Tahanaz have done this. And we got constant guidance from Dr. Nisar Ahmed, and we get several funding from different research organizations. So thank you so much.
Thank you, Professor Atiyatul Wahab, for this very nice, beautiful presentation. Now it is open for discussion. If anybody want to make any comment or queries, please.
Yes.
Microphone coming, please.
Thank you so much, Dr. Atia, for a wonderful presentation. And I see a lot of potentials of the anti-diabetic drugs. And I have a specific question regarding streptozotocin. As it is still now, it is the most popular method to prepare a type 2 diabetic model in rats and mice. But there are several toxicity reported as well. So how can we combat these two dimensions? Like, is there any alternative methods, better methods? Because you are working on it. Thank you.
Yeah, definitely. We did not check it. The other model that's we developed just this model. So maybe I can search for it and get the new model done. Yeah, but definitely there's toxicity always. But we haven't checked the other one.
Thank you, ma'am, for comprehensive talk. And we regard you as a leader in NMR. So my question is regarding the STD NMR. This is just for my knowledge, because first time I'm hearing like the interaction of enzymatic activity in the NMR. So for this study, we have to compare the NMR of power compound with the NMR of the enzyme and compound.
Okay, so you asked actually two questions. One is the enzymatic activity you would like to check using NMR. You can definitely check it, but there are some enzymes that immediately react. NMR is a slow technique. It requires time. You need to prepare, you need to stabilize sample, you need to give a pulse, you need minutes. Those reactions will just immediately happen, then you cannot observe in NMR. But if a reaction is slow enough that you can observe over minutes or hours, then you can observe. What you can do, you can have your enzyme and the reagents in the NMR tube, and then you add your substrate, and then record the proton NMR spectrum. But you should have the NMR of your substrate already with you, so that you can compare the result of the peaks. And then after you add the substrate with the enzyme and the reaction, and you know already the reaction is ongoing, then when you have to check the proton NMR. You just need a proton NMR, you don't need other. But you need to check each after, for example, 2 minutes time, 1 minute time, 5 minutes time, depends on the reaction process that you are using. You know, how fast your enzyme react with the substrate. And then you can measure the peak of the product, because as soon as the reaction proceeds, substrate will be diminishing, the peak of the substrate will be diminishing, and the peak of the product will be increasing. So with these, you have to record several NMR experiments based on the time that your substrate and enzyme require to react. And then you can compare. This is the whole process that you can easily monitor. which one is decreasing and now the substrate is increasing. That's the one question. The second question that you asked about the pure protein and the ligand interaction in STD NMR. That is different. This one is different. This is just the proton NMR. In STD NMR, you need to saturate the protein. And you do not need only the protein, but you can use the STD NMR in cell as well. And this is already been reported in literature. But definitely cells immediately settle down. So you need to do it very fast experiment so that the cells, because in NMR tube, you need to give time, first of all, and there is a region there where you can give the pulse to the NMR, not at the bottom and not on the top. There is a small region where the pulse is right to the sample. So your cells need to be there in that region. So the NMR experiment should be good. So not only the protein, pure protein, but in cell NMR you can also do. But definitely the interaction when you give a pulse either to cell, but cells you cannot find out where it is actually binding your ligand. But protein, you know that this is a protein, and then you need to have the multiple sets of NMR recorded. You need to have the STD NMR because saturation transfer difference. So the difference NMR. You record the proton NMR and then you record, you give a pulse and then you record NMR and then you subtract this. And then that is the difference NMR that you would like to observe. But you have to do it only with protein. You have to do it only with ligand because you do not want to report an artifact, right? So, and then you have to do with ligand and protein together. And whatever the earlier control experiment, you have to subtract and then you get the actual STD that you were trying to observe. Okay?
Thank you, ma'am.
Thank
The
you.
second-
Thank you. I think we are a lot behind time.
Thank you.
Madam, yeah. Yes, ma'am.
Informative lecture, really. Thank you for that. But you said that during LD50 acute test, we actually, you have correctly said that a single or multiple dose should be given within 24 hours time. But the observation goes for 14 days. You have to wait. What is happening with
No,
those?
you are right. We have done 40
But
days, 2
it's
weeks.
for 24
Yeah, it was my mistake. Yeah.
Yeah.
Yeah,
and-
it's
and
quite,
then-
it's quite
and then-
long time has been
Yeah.
passed that we have
Yeah,
done
and
this
then you
work.
can go for the second
Yeah.
part.
Yeah.
And then regarding the HbA1c level,
Hmm.
you cannot, you have to wait for at least 3 months because the lifespan of erythrocytes is 3 months. So it is not easy to make one. I think you should pay attention on that. And regarding the direct, why you go for measurement of direct bilirubin to see the liver function test, you go for testing the AST, ALT, the enzymes. It is easier because we know that the drugs and all the xenobiotics, they have got their metabolism through liver. So that is easy. Thank you.
Okay, thank you, madam.
Thank you. Thank you. I think we'll proceed to the next speaker because we are very, very behind time. And our next speaker is Fatima Anjum. The speaker shown on the program is not available. I nominated from the same institute. So it's online. This will be online. Fatima Anjum will present this online. Yeah.
Hello.
Hello, Fatma, we can hear you. Can you please
Hello,
share?
hello. Yes, I can hear you.
Okay, please share your screen, your presentation.
Okay.
She is presenting anti-diabetic and anti-inflammatory potential of hibiscus.
Is it okay?
Yeah. Yes.
Thank you.
So,
Okay, please put it on the slideshow. Yeah. Put it
Yes,
on-
yes.
Okay, please go ahead.
Good afternoon, everyone. My warm greetings and thanks to the Honorable Chair of the session and the esteemed guest for providing such an opportunity. I am Fatimanju Faruqi and I am presenting the paper on behalf of Mir Taufikur Rahman. The topic of the paper is anti-diabetic and anti-inflammatory potential of Hibiscus subzarifa fruit aqueous extract and impact on COX-2, TNF-alpha, interleukin-1 beta, and IRS-1 in anesthesia-induced type 2 diabetic rat. Diabetic mellitus is a chronic endocrine metabolic disorder which is characterized by persistent hyperglycemia due to the absolute or relative deficiency of insulin secretion, insulin action, or both. It is the fourth leading cause of death in the developing countries. Type 2 diabetes is the most common diabetes in the world, and all cases are and most of the cases are type 2 diabetes. Several risks of diabetes complication, particularly cardiovascular disease, peripheral vascular disease, coronary artery disease, stroke, neuropathy, renal failure, retinopathy, amputation, and blindness. The International Diabetic Federation estimates that 589 million adults, which is 20 to 79 years old, and are living with diabetes globally. This figure is projected to rise 853 million by 2050. In Bangladesh, according to the IDF report, nearly 13.1 million people with diabetes, which is 7th highest number globally. No effective treatment or prevention has been identified to cure this disease. Existing treatment options have severe limitations. Therefore, there is a clear need for the development of alternative strategies for diabetes therapy to delay the progression of diabetes complication. Type 2 diabetes and inflammation. Recent studies have increasingly connected to type 2 diabetes mellitus to both metabolic dysfunction and chronic low-grade inflammation. Pro-inflammatory mediators such as cyclogenesis 2, tumor necrosis factor alpha, and interleukin-1 beta are now understood to play critical roles in the disease pathophysiology. Similarly, interleukin-1 beta produce the activated macrophages and stress beta cells, promote beta cells apoptosis, and inhibit insulin secretion. These mediators are often regulated through the nuclear factor kappa B signaling pathway, which is activated by hyperglycemia, lipotoxicity, and oxidative stress, creating a feedback loop of inflammation and insulin resistance. Now, cyclooxygenase 2 is an inducible enzyme promote the production of pro-inflammatory prostaglandin and elevated in insulin-resistant tissue, exacerbating local and systemic inflammation. High level of COX-2 have been observed in some tumor, and blocking its activity may help reduce tumor growth. COX-2 inhibitors are a class of non-steroidal anti-inflammatory drug that especially block COX-2 enzyme, which is involved in inflammation and pain. They are used to treat conditions like arthritis and their type of pain, and are considered as effective as traditional NSAIDs, but often cause fewer gastrointestinal issues like ulcer. TNF-alpha is secreted mainly by adipose tissue. Macrophages in obese individual inhibit insulin signaling by disrupting insulin receptor substrate 1. phosphorylation and activating serine kinase. Drug that block TNF alpha, known as TNF inhibitor, are used to treat condition where the immune system mistakenly attack the body own tissues. Interleukin 1 beta produced by activated macrophages and stress beta cell promotes beta cell apoptosis and inhibit insulin secretion. Elevated interleukin 1 beta level are common in many chronic inflammatory conditions and can be therapeutic target to reduce symptoms. An interleukin 1 beta inhibitor is a type of therapy that blocks the activity of pro-inflammatory cytokine interleukin 1 beta. These inhibitors are used to treat various inflammatory diseases such as rheumatoid arthritis, systemic juvenile idiopathic arthritis, and cytopyrine-associated periodic syndrome. Insulin receptor substrate 1 is a key signaling protein that acts as a docking site for protein in the insulin signaling pathway. It seems mediated to metabolic effect to insulin and insulin-like growth factor 1, playing the crucial role in regulating glucose homeostasis and cell growth. It is encoded by the RS1 gene, is approximately 180 kilodalton in size, and its this function has been linked in insulin resistant and type 2 diabetics. This is the molecular pathway of insulin resistance. Our metabolic stress like obesity, high fat diet, sedentary lifestyle, it expansion the adipose tissue, and the adipose tissue secreted the pro-inflammatory cytokinase TNF alpha, interleukin 6, MCP1, and pro-inflammatory kinase can TNF alpha and interleukin 6. It's make chronic low-grade inflammation, serine phosphorylation of IRS, impaired insulin signaling pathway, make insulin resistance, and it's make hyperinsulinemia, further metabolic dysregulation and inflammation. It's make more lipid accumulation, further adipose tissue inflammation, and sustain insulin resistance. In the summary, obesity and summary of this flowchart, we can tell obesity and poor lifestyle causes fat tissue to grow too much and attract inflammatory immune cell, which release chemical that block insulin from working properly, leading to insulin resistance. And higher blood sugar, more fat buildup, and a vicious cycle that keep inflammation and diabetes getting worse. We are working on natural product in diabetic mellitus treatment. Actually, in ancient time, plants have been used for the treatment of various ailments, including diabetics. Today, 25% of prescription drugs are originally obtained from plant source because plant contain thousands of compounds. Some of them might be useful by themselves. All the conventional anti-diabetic medication have demonstrated efficacy in glycemic control. And have shown severe adverse effect. Our plant is Hibiscus sabdarifa. It's commonly known as Rosella, a member of Malvaceae family. It's widely cultivated across Asia, Africa, Latin America, including Bangladesh, India, China, Egypt, and Nigeria. Rosella widely used in the food and beverage industry to make teas, juice, jam, wine, fermented drink, and natural dyes. It's also Rosella has well-established history in traditional and complementary medicine for the diuretic, analgesic, hypotensive, and cholesterol lowering effect. These benefits are attributed to its high content of anthocyanin, polyphenol, and flavonoid, which have demonstrated powerful antioxidant, anti-inflammatory, and hypoglycemic properties. Studies suggest that Rosella may improve insulin sensitivity and reduce chronic inflammation, making it a viable candidate for managing type 2 diabetic mellitus. Now our general objective is to evaluate the anti-diabetic and anti-inflammatory effect of aqueous extract from the hibiscus subdarifa calyx on the expression of COX-2, TNF-alpha, interleukin-1 beta, and IRS-1 in NSTZ type 2 diabetic rat. Our specific objective is to perform preliminary phytochemical screening of H. subdarifa fruits aqueous extract to assess the effect of H. subdarifa fruit aqueous extract on glycemic and lipidemic parameter in type 2 diabetic model rat, to explore the role of H. subdarifa fruits aqueous extract on COX-2, TNF-alpha, interleukin-1 beta, and IRS-1 gene expression level. The rationale of the study is diabetes has become one of the most challenging public health problems of 21st century. Existing oral hypoglycemic agent and insulin have prominent side effect to fail to significantly alter the course of diabetic compliance. Plants are being used for the treatment of diabetes for centuries. So therefore, H. subdarifa may be considered one of the most important and interesting medicinal plant as the potential source that should be explored for the development of strong immune system in diabetic patient, as well as the discovery of newer and safer anti-diabetic as well as anti-inflammatory drug candidate. This is our research plan. At the end of our now method and materials, we done chemical study and biological study. For chemical study, plant sample are collected from Khagrachori Hill, and it's we collect identified number from National Herbarium. Then we sanded it and grinded it in particular mesh size. Then we aqueous extract was aqueous extract made followed by a maceration technique. Filtration was completed by Hartmann filter. Hartmann #1 filter paper. Sample was evaporated by rotary vacuum evaporator, and finally drying process was completed by freeze drying. This is the process we standard procedure we done in our lab. Now, biological study, 28 hours long, even rat pups was injected streptozotocin in citrate buffer at the dose of 90 milligram per kg body weight per 10 ml. 3 months later, when they become adult, oral glucose tolerance test was done to check their glycemic status. Blood sample was collected by tail cut method consecutively after 30, 60, and 120 minutes prior from the time of the first blood culture under halogen anesthesia. Our experimental design, we make 4 groups for one is a normal water control group, and second is type 2 diabetic model rat. Third one is we treat type 2 diabetic model rat with glycoside was administered, and fourth one is hibiscus subdarifa fruit aqueous extract treated group. Route of drug administration for this study, glycoside and hibiscus subdarifa administered orally for 28 days, and we collected blood sample and we After 28 days, we collected muscle, adipose tissue, and liver tissue, and we dipped into triazole reagent for further investigation. This is the biochemical analysis we done for this research, and we the molecular gene expression we done with by Studio RT-PCR. Now result and discussion. For chemical study, in preliminary phytochemical screening, we found tannin, saponin, flavonoid, and phenol are present in my extract, and others are absent. Now, the effect of hibiscus subdarifa food calyx extract on fasting serum glucose level of type 2 diabetic model rat. The chart showed that our extract treated rat 30% decreased their fasting glucose level compared to the baseline value. The group and the TZ and cholesterol Rosella treated showed that 2% decrease significantly. TZ TZ is significantly decreased 2%, where the drug treated decrease 1.78%. And the cholesterol, it also decreased significantly, where drug decreased 5% of the baseline value. And here also HDL value is significantly increased, and the LDL value is significantly decreased. Now, effect of hibiscus subdarifa food calyx extract on molecular gene expression level in type 2 diabetic model rat. We calculating by delta CT value against our housekeeping gene GAPDH and beta actin gene. And we get that the COX-2 TNF alpha and interleukin 1 beta is decreased significantly compared to the control group, so that we can tell our extract can reduce inflammation. And here the insulin receptor substrate 1 is increased significantly compared to the control group. So Rosella can effect on insulin production and reduce insulin resistance. Now, the conclusion is the study demonstrated that the aqueous extracts of hibiscus subdarifa fruit calyx contain an abundant variety of bioactive phytochemicals, including tannin, saponin, flavonoid, and phenolic compound. Its anti-diabetic potential may be attributed to its ability to lower serum glucose level and improve lipid profile. This effect appears to be mediated by the upregulating of interregulating IRS1 gene expression and suppression of COX-2 TNF alpha and interleukin 1 beta gene expression in the liver, muscle, and adipose tissue of anesthesia-induced type 2 diabetic rat. Based on this finding, it may be hypothesized that A. subdarifa could emerge as a promising novel anti-diabetic and anti-inflammatory therapeutic agent, warranting further details investigation. This is the reference we used, and I would like to express my deepest gratitude for Professor Dr. Begum Rokeya, Dean and Faculty of Basic Science, for her guidance and encouragement and support throughout the course of the research. And I'm thankful to ANRAP and BUHS and I am also deeply grateful to Professor Dr. Shahinul Haq Khan, MD Shahjahan, MD Shalom, MD Helal, MD Yasin for their dedicatedly support in our research. Thank you.
Well, thank you for giving the good news about this rosella, because rosella is widely used as a tea in many parts of the world. So anyhow, I think we are very behind time. If you have got any question or clarification. Okay, okay, we proceed to the next presentation then. The next presentation will be online from Egypt. Egypt, I don't know which one will be presenting. Sameh Abu, Dr. Sameh Abu Ji and Professor Osama Muhammad Ahmed, who will present it, and they will be talking on anti-hyperglycemic effects of some Egyptian plants and algae. Algae or algae.
Oops. Question করেনি তাই না ভাইয়া খালি
Hello, good morning. Can you hear me?
Yeah.
Okay. Assalamu alaikum. Can you hear me well?
Alaikum assalam. Please proceed.
Yes, thank you very much. Let me first thank His Excellency Professor Dr. Muhammad Iqbal Chaudhary for giving me this opportunity to present part of my work today, along with my colleague Professor Osama Muhammad Ahmed, who will present half of the work we have done on the anti-hyperglycemic effects of some Egyptian plants and algae. I'm a professor of medicinal plants, head of pharmacognosy department, faculty of pharmacy, Bani Suef University in Egypt. And in Egypt, there was the first referral to diabetes, where in this papyri, as you can see, in the left hand of this engineer who is named Amenhotep, a brilliant engineer actually, who was behind the first stone building on earth, the stepped pyramid in Egypt. He was not only a brilliant engineer, but also he was a brilliant physician, and he in his hand he wrote this papyri, which included the first referral to diabetes in the form of incurring urine, which considered first referral to this disease, which is a great burden nowadays, 3,500 years ago. Nowadays, according to the International Diabetes Federation, in Egypt, with its 600 million adult population, there is a prevalence of 22.4% of diabetes among the adults in Egypt. As you discussed yesterday, diabetes is a heterogeneous endocrine disorder in which the hyperglycemia is a common feature. There are two main types, either the insulin dependent, which is called type 1, and usually it's juvenile insulin, and the type 2, which is called non-insulin dependent diabetes mellitus, and it's of adult insulin. For the therapeutic approaches for the two types, the insulin dependent usually is treated with insulin with some adjustment in diet or exercise. But today we focus our work on non-insulin dependent diabetes mellitus, in which there is a significant concentration of insulin, but or lack of insulin. In case of the high concentration of insulin, as you all know, and as you discussed in the presentation today and yesterday, there is a resistance of the cells to use this high concentration of insulin. There is a need for higher concentration of insulin to give the same response as in the normal cell. Here we use the oral hypoglycemic agents to increase the insulin secretion, to increase the peripheral glucose uptake, or to decrease the glucose absorption, or decrease the hepatic glucose production. The aim of our work actually was to find novel treatments for plant or marine origin in the Egyptian environment, which possess potential therapeutic properties for especially type 2 diabetes with no or fewer side effects, so that they can be used instead of the oral hypoglycemic agents. This is the plan of our work. It began with collection of plants and algae and taxonomical authentication and deposition of the voucher specimen. Collection of these plants was done, and then it was air dried, grounded, and extracts were prepared from these plants or algae. The dried extracts were kept under minus 20 degrees centigrade until biological evaluation, then subjected to simple preliminary phytochemical screening, then the toxicity screening and the evaluation of the anti-hyperglycemic activities using nicotinamide streptozotocin type 2 diabetes male albino mice. The first step was the collection of the plants, and especially the algae, which were the hard step actually to collect. We moved from Banis Wave, which is here in Egypt, Middle Egypt, Upper Middle Egypt, to a city called Qusayr on the Red Sea of Egypt, almost 600 kilometers by car. And then these are some of our team members in the scientific group. We were working together to collect the algae like Caulerba lantifera and Alva lactuca. The second step was to dry these algae and get the powder and extract them with 80% aqueous methanol. These are the yield which we got from the plants used, 16 plants and 4 algae. All of the plants were extracted with, as a common protocol, 80% aqueous methanol, except for Plantago psyllium, because we found some reports about the effect of mucilage from Plantago psyllium on the diabetic mice. So we prepared the mucilage part of these seeds of this plant using the usual protocol. Then we made a simple phytochemical screening using the unspecific, actually, chemical test, but we wanted to identify the major phytochemical classes which are present in these extracts. This table are showing you the result of this simple color test phytochemical screening. Then we proceeded to the toxicity screening of these plants. All of the plants were considered safe at very high dose, 2,000 milligrams per kilogram body weight, except for Schicorium intybus, which show one of the animals was dead at this high concentration. So we calculated the LD50 for this, especially for this extract of Schicorium intybus. And this table is showing you the determinations which we got. The LD50 was determined, and then the therapeutic dose which was used in the male albino rat mice, was also calculated. We used a diabetic model depending on nicotinamide and streptozotocin, 120 milligram per kilogram nicotinamide and 200 milligram per kilogram body weight streptozotocin to induce diabetes in the mice. And then the extracts were administered in the mice using gastric tube at 2 doses, 10 milligram per kilogram body weight and 50 milligram per kilogram body weight for 1 week. Then the blood samples were collected from fasting and glucose-loaded mice, and then the serum glucose level was determined. The results are shown in the next tables. As you can see, these are the normal or the negative control mice. These are the diabetic control mice where we induced diabetes using, as I said, nicotinamide and streptozotocin. And these are the result of the plants like Bauhinia variegata, Bougainvillea spectabalis, Cassia acutifolia, Chicoryum intypus. This is the part used. Here we use the leaves. L donates the leaves. F is the fruits. And this is the dose, 10 milligram per kilogram body weight or 50 milligram per kilogram body weight. And this is the result of the fasting animals and the other one after glucose upload. Cassia acutifolia was very high in both cases at the both dose, the 2 dose levels. These are the other plants like Cucurbita pepo, Eucalyptus globulus, Fraxinus orinus, Mangifera, Morus alba, Morus nigra, Optionia ficus indica, Plantago psyllium, Salix aegyptiaca, Serenua ripanis. All of these plants are common in the Egyptian, but especially Cassia was highest among the plants. Among the algae, we used 4 algae: Caulerpa lentiflora, Enteromorpha intestinalis, Spirulina vesicularis, and Alva lactube. Because of the Cassia acutifolia was the highest, we made a phytochemical characterization of this plant using the preparative chromatographic work to isolate the main constituents using a thin layer chromatography, column chromatography, and preparative HPLC to obtain the main compounds in the active extracts. And these were shown to be chrysophenol, alloimodine, ryein. These are anthracene aglycones, and sinucide B, anthracene glycoside, among a very, and also the common flavonoid aglycone camphor. Interestingly, when we made a literature review, we find other Cassia species like Cassia angustifolia, Cassia orculata, Cassia glauca, and Cassia siamia, also shown in different reports a good response against the hyperglycemia associated with diabetes. So in conclusion, anti-diabetic effects of 16 plants and 4 algae were evaluated for treatment of diabetes mellitus. All of these are common in the Egyptian environment. The highest result was for Cassia acutifolia leaf extracts, giving us the highest potential as a herbal medicine for diabetes. A potent anti-hyperglycemic effect associated with non-significant increase in serum insulin level was demonstrated for Cassia, Fraxinus, Salix, Plantago, Serenua, and Bougainvillea species. A potent also anti-hyperglycemic effect associated with insulinotropic action was demonstrated for Eucalyptus, Cucurbita, Morus nigra, Chicoryum intypus, and Sonchus oleracea. All of the tested algae have anti-hyperglycemic effects at higher degree without a significant increase in serum insulin concentration. I have to acknowledge the project which we supported us in this work, which we obtained from the Science and Technology and Innovation Funding Authority in Egypt, the main funding body for research, under the title Search for Novel Anti-Hyperglycemic and Anti-Hyperlipidemic Plant and Algae Constituents from Egyptian plants and algae, under the category of basic and applied research. We worked together with magnificent, actually, researchers under the supervision of Professor Dr. Muhammad Badr Ashur and Professor Dr. Usama Muhammad Ahmed. We were the PI and co-PI of this project, and the late Dr. Rasha Ahmed, Dr. Ayman, and Dr. Ihab Abdullah. I have to acknowledge all of them and thank you, and give the floor now to Professor Usama Muhammad Ahmed, my colleague, to share with you the other part of the work in which he is actually an expert in testing the anti-diabetic and anti-hyperlipidemic activity and understanding the mechanism behind this activity. So I leave the floor to Professor Usama to give his input in this project. Thank you very much.
On the call upon Professor
Osama.
Assalamu alaikum. Do you hear me?
Yeah, we can tell.
Bismillah ar-Rahman ar-Rahim. First, we are very grateful to Prof. Dr. Shodari and this conference organization committee for giving us a chance to present some of our works. In this presentation, we will talk about the anti-hyperglycemic effect and the mechanism of action of one commonly used plant in traditional medicine, which is Balanitis aegyptiaca, which is used by many of diabetic populations. And one algae, which is green macroalgae, Olfa lacteuca, as an example of green macroalgae. We used oral glucose. We usually used the oral glucose tolerance test and fasting glucose and fasting glucose and postprandial glucose to screen the hyperglycemic state and to assess the anti-hyperglycemic effect of the tested plants or algae. After confirming the anti-hyperglycemic effect, the modes and the mechanisms of action are proposed by assessment of the effects on intestinal glucose absorption in situ, anti-glucosidase and anti-amylase activity in vitro, insulin release from the isolated islets of Langerhans in vitro, hepatic glucose output in vitro, peripheral glucose uptake by the isolated diaphragm in vitro, pancreatic islet histological changes and the serum insulin and the C-peptide levels, insulin granules detected by immunohistochemical techniques, detection of liver glycogen phosphorylase, and glucose 6-phosphatase, insulin receptor, and the various mediators involved in signaling pathway of the insulin action, such as insulin receptor substrate 1, insulin receptor substrate 2, and BI phosphoinositol 3 kinase, and so on for other mediators. Peripheral tissues, glucose transporter 1 and 2, which are responsible for entrance of glucose into cells of the peripheral tissues. First, we talk about the anti-hyperglycemic mechanism and mechanisms of action of Balanitis aegyptiaca, which is growing in Egyptian, which grows on rocks which is grown in deserts and in arid and semi-arid regions of Africa, Middle East, and South Asia. The experimental design of this experiment and the animal grouping, the rats was the type 2 diabetes mellitus was induced by injection of streptozotocin at dose level 60 milligram per kilogram after 15 minutes of injection of nicotinamide. After 1 week or 10 days, the glucose was screened for the hyperglycemia, and the diabetic rats were resubdivided into diabetic control which is given the vehicles. Diabetic group treated with fruit aqueous extract at dose level 200 milligram per kilogram. Diabetic group treated with seed aqueous extract at the same dose. This is the detail for the animal grouping. The results. This figure shows the oral glucose tolerance test of different groups. The diabetic rat exhibited significant increase in glucose level at different time intervals. The treatment with Balanitis aegyptiaca, fruit and aqueous fruit and seed extract, produced potential and significant amelioration of the oral glucose tolerance curve as indicated in the figure. We also detected serum insulin and C-peptide level. Both serum insulin and C-peptide level exhibited a potential decrease in diabetic rats. The treatment with fruit and seed extract produced a significant amelioration and improvement of serum insulin and C-peptide levels. We calculated HOMA beta cell function, HOMA insulin resistance, and HOMA insulin sensitivity from fasting insulin and fasting glucose by different equations. The diabetic rat exhibited significant decrease in HOMA beta cell function, and the treatment with both fruit and seed extract produced a significant increase in the HOMA beta cell function. In association with these changes in beta function, we perform histological, perform histological section. The histological section of the diabetic rat exhibited a significant decrease in the size and the number of islet cells within the islets of Langerhans, as shown in C and D. These are sections, histological section of pancreas of diabetic rats. The treatment with fruit and seed extract produced a marked increase in the islet size and increase in the number of beta cells within the islets of Langerhans. HOMA IS and HOMA IR were also calculated. The diabetic rat exhibited significant decrease in HOMA IS and a significant increase in HOMA IR. The treatment with fruit and seed extract exhibited significant increase in the HOMA IS and a significant decrease in the HOMA IR. We also detected liver glycogen and glucose 6-phosphatase and glycogen phosphorylase. The glycogen content in the liver significantly decreased in the diabetic rats and increased by treatment with the fruit and seed extracts. Glucose 6-phosphatase and the glycogen phosphorylase was increased significantly in diabetic rats, and the treatment with either fruit or seed extract produced a significant amelioration of the glucose 6-phosphatase and the glycogen phosphorylase. The glycogen phosphorylase is the key enzyme for glycogenolysis, and it was increased in association with the decrease in the glycogen content. Glucose 6-phosphatase, which is responsible for release of free glucose, which will be ready for release from the cells into the blood. Insulin receptor insulin receptor subunit was detected by detection of the messenger RNA expression, and it was not significantly affected in diabetic rats. It was only significantly increased as a result of treatment with the fruit extract. This figure summarized the effect of both fruit and seed extract of Balanitis aegyptiaca. The treatment with fruit and seed extract produced improvement in insulin secretory action, and this was evidenced by increase in serum insulin, increase in serum C-peptide, increase in HOMA beta cell function, and increase in the number of beta cells as indicated in the histological section. The treatments also induced increased insulin sensitivity and reduced insulin resistance, and this was evidenced by decrease in HOMA-IR, increase in HOMA-IS, decrease in free fatty acids, which have important role in insulin resistance, increase in adipose tissue insulin receptor, especially with the fruit extract. Another data which is not included are the oxidative stress and antioxidant defense system parameters, which include glutathione peroxidase, lipid peroxidation, and SOD. The lipid peroxidation was decreased by treatment, while the antioxidant substrate glutathione and the antioxidant enzymes such as glutathione peroxidase and superoxide dismutase were increased as a result of treatment. In conclusion, the Balanitis aegyptiaca fruit and the seed extract have potential anti-hyperglycemic effect, which may be mediated through improvement of insulin secretory response, through increasing the beta cell function and the number, and amelioration of insulin action. The second tested algae is Olfa-lactuca. The Olfa-lactuca also known as sea lettuce, is a green macroalgae. It is belonging to chlorophyta. It is found attached to rocks near the seashore and is commonly found in both Red Sea and the Mediterranean Sea. The experimental design.
I think we are too behind time. If you could
Yes.
shorten your
there is two
Come to
another
conclusion.
slides.
please.
Yes,
Come to the
another
conclusion.
slide,
please.
and yes. The Olfa-lactuca, this is the experimental design, normal control and diabetic rats. Diabetic rats were subdivided into diabetic control, diabetic group treated with olvan infusion, or Olfa-lactuca aqueous extract, diabetic group treated with olvan polysaccharides.
Professor Ahmad. Professor Ahmad.
Yes, I will go to conclusion.
Come to the
Okay.
conclusion. Please,
Yes.
please come to the conclusion.
Yes. Olfa-lactuca aqueous extract in the form of infusion and olfan polysaccharides have anti-hyperglycemic potential, which may be mediated via reducing intestinal glucose absorption, increasing the beta cell function number and function, enhancing peripheral glucose uptake, and decreasing hepatic glucose output. Thank you. This is the references.
I have written announcement. We shall have one more presentation and have the last
Yes.
presentation after lunch.
Yes.
So may I request Fakir Sharmin Srishti. She is from Haji Danesh University of Science and Technology, that is in the northern part of Bangladesh. And you have 15 minutes to present it.
15 minutes.
Assalamu alaikum, sir. Can you hear me?
Yeah.
Okay. Thank you, sir, for your lovely introduction and giving me the floor. Assalamu alaikum and a very good noon to everyone present. It's an honor to join this streaming gathering virtually at the 8th ANRAP and OIC Comstek Regional Seminar 2025. I'm Fakir Sharmin Srishti from Department of Chemistry, Haji Muhammad Danesh Science and Technology University, Dinajpur, Bangladesh. My research focuses on the chemical and biological investigation on some edible culinary species at cumin, black cumin, and fenugreek. Okay, let's start with the first slide. First, from the introduction, actually, we all know that the culinary spices are a variety of aromatic substances derived from seeds, bark, foods, and they are used in cooking for enhance the flavor, aroma, and appearance of food. Also for their nutritional value, health benefits, and preservation. So, among these, I have selected three spices. They are cumin, black cumin, and fenugreek. In South Asian countries, they are known as jeera, kalo jeera, and methi, respectively. So let's go to the next slide. It's the review of literature.
Yeah, let's sit
Actually,
like that.
according okay, so according to the International Diabetes Federation, since it's 13.1 million adults are actually affected by diabetes in 2021. That's why I have taken this cumin, black cumin, and fenugreek that demonstrate therapeutic potential and including hypoglycemic and anti-hyperlipidemic, anti-cancer, and antibacterial properties. So the objectives of my research are to screen the glycemic effect and lipidemic status, and to assess the cytotoxicity of this extract and investigate the antibacterial activity by following the well diffusion method. Beside this, as a chemist, my main target to isolate the bioactive compounds and characterize them by following the spectral analysis. So next, move to the next slides, and these are the features of my research work as materials and method. I have designed my research work in sample extraction, anti-diabetic test, cytotoxicity test, antibacterial test, isolation, and chemical studies. So, for the further, based on these astonishing bioactive results, the ethanol extract of cumin and black cumin was further isolated by following this schematic diagram, like using with the help of solvent increasing polarity, that is n-hexane, dichloromethane, ethyl acetate, methanol, etc. The fractions are then concentrated by following the evaporation using the rotary evaporator. And for the
Can you quickly,
okay, okay, sir.
Dr.
So I'm going through the
Srishti,
result and discussion. Yes, sir.
Srishti, listen to me. Can you move the slides? Can you, your slides are not moving. Or if you don't have slides,
Actually, I'm moving here. Okay, I'm going back, sir.
okay, go ahead.
Can you see now?
Yeah, that's right.
Okay. Sir, can you see it now?
Yeah.
Okay. Okay, now it's time for revealing the most captivating part of my research work. Actually, we know that in diabetic conditions, excessive weight gain, and particularly from fat, may worsen the insulin resistance. Here we can see the significant loss of body weight for the ethanol extract of cumin, that is 30%, where glycoside reduced it only by 16%. But the weight was reduced increased in the case of the normal and diabetics water control group. And in case of oral glucose tolerance test, the glycoside increased the postprandial serum glucose only by 13%. And surprisingly, chloroform extract showed the acute effect, that is 99%. Both ethanol and chloroform extract of fenugreek showed significant increase in 49% and 38% respectively. So in my next slide, we can see the fasting serum glucose level and lipid profile for triglyceride level and cholesterol. Here we can see that the ethanol extract of cumin 22% and the chloroform extract of black cumin 35% are most effectively for reducing serum glucose level. So we can say that black cumin extract performed better than that the standard drugs. And now for the fenugreek extract and the fenugreek extract of chloroform, that is 20%, and ethanolic extract of the black cumin and cumin demonstrated the remarkable reduction in the cholesterol level, that is 15%, where the glycoside reduced only by 2%. And all the chloroform extract showed the significant values by decreasing the triglyceride level too. In my next slides, you can see the lipid profile for HDL and LDL level test. Here we can see the cholesterol level for ethanol fraction. Black cumin showed the highest value for increasing 11% HDL as glycoside, but again with the cumin showed the maximum decrease of LDL, that is 17%, whereas the glycoside it's decreased only 4%. So on the other side, we can see the chloroform extract of black cumin and fenugreek showed the astonishing increased HDL by 98% and 73%, beside decreasing the LDL, that is 42%. So we can say that black cumin and fenugreek chloroform extract showed the strongest cardioprotective effect. And in the liver glycogen estimation, surprisingly, we observed a remarkable result for ethanol extract of cumin on liver glycogen level increasing by 200%. Of type 2 diabetic model rate, and black cumin aqueous extract that is 128%, and cumin aqueous extract that is 102%. Now, in this case, we can say that the better glucose storage capacity was shown by them. Okay, so now the cytotoxicity test, the cytotoxic activity of the crude extract of cumin, black cumin, and fenugreek were demonstrated. While aqueous extract, we can see that the fenugreek showed the highest amount, that is the most anti-cancer activity with 0.88. And beside this, the fractions of ethanolic cumin and black cumin showed the tremendous the cytotoxicity, which is greatest in the ethyl acetate one. Now, in case of antibacterial activity, we can see that the food extracts was demonstrated again a gram-negative bacteria called Aromonas hydrophila. All of the samples showed effective zone of inhibition. Among them, cumin extract and its ethanol extract showed the maximum one, that is 35 millimeter, and further the fractions of the ethanolic cumin and black cumin showed the tremendous antibacterial activity, that is highest one for the ethyl acetate here also. And this is the table. I won't show this, and it's the schematic diagram where I have isolated the single spotted, but I'm going through my chemical study now.
Charmaine, can you
Yes, sir.
come to the conclusion? Can you come to the conclusion?
Yes, sir, of course. I'm going now. And I want to share this one because a single spot, that is a cuminaldehyde, I have isolated here because it's so tough to isolate the single compound in our laboratory with less facilities. So this is the cuminaldehyde where I can isolate it by following that carbon oxygen stretching in 1701. And with the NMR and NMR with proton and carbon 13 NMR, I have finalized then characterized it's like cuminaldehyde, where PubChem also confirmed it. And I have also given here some tentative structures, and I'm not saying anything about it. Okay, my conclusion. Okay, extracts of the cumin, black cumin, and fenugreeks, they showed the potential for the managing diabetes. So they also showed the antibacterial and anti-cancer activity test. That's why finally we can say that extract will be regulating the HDL, LDL cholesterol level and preventing cancer and bacterial disease. So my future study will be, since I have isolated the pure cuminaldehyde, so synthesis of the derivatives like seed base from cuminaldehyde to explore its potential application with their computational study, and preparing nanoparticles from them with the extracts of cumin, black cumin, and fenugreek with their comprehensive biological and chemical study will be my next target. So before finishing my presentation, I'd like to give my heartiest thank to UNRAP and 8th OIC, and to my honorable supervisor, Professor Dr. Balaram Roy, and co-supervisor Dr. Uttam Kumar Sarkar sir. And beside this, I would like to thank Professor Dr. Begum Rokia from Bangladesh University of Health and Science for her assisting in determining the glycemic and lipidemic status. And have a glance to my HSTU campus. Thank you, everyone. Thank you for listening.
I think we'll go for lunch and then come back. Is it all right if we come back at 2 and have the last presentation? Okay, thank you. Right. So, yeah. Well, I think we start the session, but otherwise the next session will be even later. This is the last paper of this session, and this will be presented by Mr. Tanvir Mahatab Rifat, who is a research fellow of ANRAP at the Bangladesh University of Health Sciences, and he is going to present antioxidant and anti-diabetic effects of Synacanthus mutans leaves in streptozotocin induced type 2 rash. Rifat, please. Is he here?
Tanvir,
Yes,
Tanvir,
sir.
please come. It's your turn.
Yes, sir.
Tan Hold on. This one. Oh. Oh, yeah.
Assalamu alaikum.
Walaikum assalam. Okay, Tanvir, put your slides on the screen show. Okay, we can see. Please continue.
Tanvir, please start.
Assalamu alaikum. Good afternoon.
Alaikum
Thanks to
salam.
my honorable chair of the session and the esteemed guest for providing such an opportunity. I am Tanvir Mahatab Rifat, and my presentation topic antioxidant and anti-diabetic effect of Synacanthus mutans leaf in anesthesia-induced type 2 diabetic rat. Role of growth for and insulin receptor gene expression. My previous presenter told about the discuss of diabetes, so I skip this. Now I am discuss about about molecular mechanism of growth for an insulin receptor. Insulin secreted from insulin and bind to the insulin receptor, and growth for channel often glucose transport to the cell membrane, and glycogen synthesis of glycogen, glucose to glycogen, and glycolysis procedure, glucose to pyruvate, and pyruvate to fatty acid. Natural product. Since ancient time, plant have been used for treatment of various ailments, including diabetes. Synacanthus nutans. Synacanthus nutans is a flowering plant native to Southeast Asia, especially in Malaysia, where it is traditionally used for its medicinal properties, known as Saba snake grass, Beraligaza, or Akorkusin. Researchers revealed that nutans contain a variety of phytochemicals, including flavonoid, alkaloid, cytoterpenoid, steroid, phytosterol, and glycoside. According to the previous study, nutans may have various health benefits, including anti-diabetic, anti-inflammatory, antioxidant, and anti-cancer properties. Moreover, flavonoids promote the growth for and insulin receptor translocation by activating MPK pathway, and their integral role of management of diabetes complication. Rationale of the study. Diabetes has become one of the most challenging public health for the 21st century. Exciting oral hyperglycemic agent and insulin have the prominent side effect and fail to significantly alter the course of diabetes complication. Plants are being used for the treatment of diabetes for centuries. Plants are the good source of the large number of secondary metabolites like polyphenol, flavonoid, etc., which may potent of antioxidant effect. Cynotans is the medicinal plant with many effective antioxidants in which we can search read compound for diabetes. Objective, general objective and specific objective. General objective to investigate the antioxidant and anti-diabetic activities and the role of growth for and insulin receptor gene expression of cynotans leaves on anesthesia-induced type 2 diabetic model. This is standard research plan of my research work. Method and materials. Chemical study. Place of study, Department of Chemistry and Pharmacology, Bangladesh University of Health Science. This is standard procedure for collection and preparation of extract. Phytochemical analysis. Dried extract was boiled with 30 ml distilled water for 5 minutes in a water bath and 3 grams of sinotans was filtered while hot. 10 in saponin alkaloid, flavonoid, phenol, estrogen and terpenoid was done by following standard protocol for preliminary phytochemical analysis. Antioxidant study. Antioxidant activity was determined by DPPS method. Biological study. Preparation of type 2 diabetics model rat. 48-hour-old rat, 40-old wrong events rat pups were injected histotoxin in cited buffer at the dose of 90 milligram per kg of body weight per 10 ml. 3 months later, when they are become the adult rat, oral glucose tolerance test was done to check their glycemic status. Blood sample was collected by tail cut method consequently after 30, 60, and 120 minute period from the time of the first blood collection under Hamilton anesthesia. Experimental design. This equation is rodent sample size calculation. According to the equation, the sample size was selected as 6 animal per group since the experiment was carried out in 4 different groups: normal control, diabetic control, GT, and sinotans. Route of the drug administration and collection of blood sample and tissue. Route of the drug administration. For pharmacological study, the drug gliclazide was administered orally at dose of 20 milligram per 5 ml per kg body weight, and sinotans leaf extract was administered orally 1.25 gram per kg body weight to type 2 diabetics model rat. For the control group, deionized water was administered 10 ml per kg body weight orally. Study period 28 days. Collection of blood sample and tissue. Biochemical analysis. Serum glucose was measured by glucose oxidase method. EG was measured by enzymatic colorimetry method. Serum total cholesterol and HDL measured by enzymatic colorimetric method. And liver glycogen was measured by Anton sulfuric acid method. Insulin was measured by ELISA method. Total superoxide dismutase was measured by WST1 method. Beta-actin gene was used for internal control, mRNA extraction, cDNA preparation, and RT-PCR analysis. Beta-actin, this beta-actin IR and growth for sequence of primer sequence. Statistical analysis was performed using statistical patterns of social science. Result will be expressed as mean plus minus standard. Statistical evaluation of data will be performed by using one-way analysis of variance and peer sample t-test. The level of significance was considered as p greater than 0.05. The molecular gene expression level was analyzed by delta-delta CT method and ethical consideration. Result and discussion. Chemical study. Preliminary phytochemical scanning. Tannin, saponin, flavonoid, phenol, and terpenoid present, and alkaloid and steroid absence. Antioxidant activity. Sinotans group IC50 value 0.98 milligram per ml and control ascorbic acid 0.591. And ascorbic acid equivalent antioxidant capacity 511.99 milligram per 100 gram of extract. Body weight. Normal control 24% increase, and diabetic control 70% increase, and GT 19% increase, and sinotans 9% increase. Fasting serum glucose level. Sinotans was decreased compared to the control group and compared to the baseline value. TG and TC level significantly decreased compared to the control group and compared to the baseline value. HDL significantly increased and LDL significantly decreased
compared to the control group and baseline values. Hepatic glycogen content, liver glycogen significantly increased compared to the control group. SOD, superoxide dismutase, SOD value significantly increased compared to the control group. Insulin level significantly increased compared to the control group and compared to the baseline value. Growth for muscle and adipose tissues significantly increased compared to the control group, and IR muscle and adipose tissues significantly increased compared to the control group. Conclusion. The finding of my study suggests that sea newtons leave extract in rich in phytochemical and exhibit significant antioxidant activity, which may contribute to improve glycemic repeat profile, SOD, and insulin in type 2 diabetic rat. The potential, the molecular and the mechanism molecular underlying these effects involves the upregulation of insulin receptor and glucose transporter type 4 gene expression in adipose tissue and skeletal muscle following the treatment with Newton's sleeve aquaceptor. Reference and acknowledgement. I would like to sincerely thank all of my teachers and gratefully acknowledge and for the financial support. I cordially acknowledge the contribution of all the fellows of NRAP, my juniors, and the lab staff of the Department of Pharmacology and Chemistry. Thank you.
I think we have got 2 minutes time. If anybody wants to inquire something or want to make comments, please go ahead. Since there is no query, it remains for me to thank the speaker, Rifat, for making a very clear and concise presentation. Thank you all.
Okay, the next session that is chaired by the Professor Dr. Vijay Kumar, and he is the Professor at Department of Chemistry, University of Peradeniya, Sri Lanka. Thank you.
Thank you, everyone. We have got 5%. Huh? Okay. We have got 5 presentations, of which one is not online, and the one which is, we start with the first one. We start with the online, non-online presentation. Dr. Mohammed Akhtar Khan, Chief Scientific Officer from National University of Medical Sciences, Pakistan. Dr. Khan, you have 15 minutes.
Bismillahirrahmanirrahim and Assalamu alaikum. First of all, I would like to thank all of my seniors, colleagues, and all the participants who gave very good talks. And before lunch, they feed us a high dose of nutritory phytochemicals. And thanks for Comstech to re-energize us. Anyhow, the rise of herbal medicine, I will go a little bit quickly. The most important factor that gives it is the dissatisfaction of the conventional medicine and the side effects of the conventional medicine. Now, the most important topic is the scientific validation and research. All we have talked is the plant medicine, but where is the clinical scientific validation? In COVID-19, there are a few examples which are a lighter note, a very good note for the use of herbal medicine like honey, ginger, kalonji, used widely as households and by practitioners and gives a very good results. Many local clinical practices showed very improved recovery times. And one of the very good example is traditional Chinese medicine. They use Qingfai for COVID-19 and they improve fever, cough, dyspepsia, and they use for whole 2 years, 2020 to 2022. And the evidence they produced was reduced progression to severe disease, especially in COVID-19. Now, if we see the reality also toward the use of medicine where we are lacking, we got a few disasters in the past. Aristolic acid, which caused 100 women with irreversible kidney. This was just because of the wrong or misidentification of the true type of the herb. High incidence of upper urinary tract cancer later, the disaster of lack of safety and toxicity data. Second one is Kawa. More than 100 cases of liver failure reported. Required liver transplant in several patients. Banned temporarily in Europe, and the reason was non-traditional use of extracts. Another example, which is banned by USA in 2004 by FDA, the use of ephedra. The high dose of ephedra caused dozens of deaths due to heart attack, stroke, arrhythmia, and thousands of adverse cardiovascular events. The most latest disaster is happened in Japan in 2024, the red yeast rice scandal. In the same case, reported 80 deaths, more than 500 hospitalized, and the contamination caused acute kidney injury. Now, here it comes. There are two things we have to consider. This is publication data of PubMed. 2022 and 23, 19,500, particularly publications on isolation and use of ingredients, herbal ingredients to any of the ailment. And 2024 and 2000 is going on. But out of all these publications, there is not a single example that has been converted into the drug that is scientifically, rationally approved. The three main causes, what we know is a lack of standardization. Every time we get an extract, every time it may have different constitution. So in our research, we must have to add the standardization that the extract at least within the 80 to 120% of the time of the percentage frame. So we can claim that throughout our research, the sample was standardized. Insufficient clinical evidence. The main basic point. We all work in vitro, in vivo, animal models, work the actual drug, either it's a whole extract, either it's a constituent, either it's identified any particular component. We need sufficient clinical evidence to call it as a drug. The other aspect is drug-drug interaction, herb-drug interaction, herb-food interaction. We perform a chronic autoimmune and insulin resistance challenge is the psoriasis, which is mostly concluded as related with the type 2 diabetes mellitus. We perform clinical trials for diabetes type 2 and psoriasis. But before reaching the clinical trial, there is a long history or length achieve that landmark. We validated our samples by 3 methods. We use phenogreek and we validate the samples with the quantification of diazosin. Just from the start of the sample till end of the sample, the whole of the trial, the whole sample was being stabilized in -80 freezers, and even the analytical validation was done by thawing and unthawing the sample 3 times. That what might be the impact of freezing or storage of the sample. Then from the first day till end day of the trial, we again validate the sample that it is intact or stable, or there is something chemical reaction is going inside. So you have, again, if I go back, so you have to standardize the sample. Once you prove, you validate that your sample is standardized, having the same constituent, then you can prove for which ailment you need to use it. Now the next is the clinical evidence. We make a study, assess changes in biochemical markers, evaluate in IL-6, TNF-alpha and CRP markers, CBC, and compare the effect between diabetic and non-diabetic psoriatic patients. We have a lot of data, but in the slide, very few data I have arranged because we are in the process of patenting the gel we have made for the whole process. The clinical trial is registered with WHO. Registry REBAC. Design is comparative interventional study, ethically approved, 200 participants, 100 were diabetic and 100 were non-diabetic. This is a human trial, proper human trial, phase 2 trial. Because before coming to phase 2 trial, we have a lot of in vitro, in vivo, animal studies, stabilization, analysis, we've done all before coming to it. Then the intervention was 50 milligram per day oral 8 weeks dosage of phenogreek extract, and we make a nano suspension gel for the psoriasis. And you will be amazed with the last slide. Anti-diabetic, fasting, and random blood, because these all the parameters, IL-6, CRP, AST, ALT, liver profile, complete blood counts that needs to be checked during the whole trial. And the proposed mechanism was where the phenogreek effects in producing the insulin or reducing the sensitivity or decrease the blockage of insulin. This was the proposed systematic diagram. What we found, the significant reduction in blood sugar, both in fasting as well as BSR, and statistically approved that the results were significant. The liver function test here, the ALT is slightly moved upward, which is a caution for us that we read or we require monitoring while dosing this supplement to the volunteers or our patients. Yeah, please. Thank you. And hematological parameters, lymphocyte, and all these MCV neutrophils have a very good significant results and a strong correlation between all these results. This was the psoriasis patient, 42 years old male, and I was very happy when he came to me and told me that, doctor, I slept very well after 14 years. Because the itching goes, there was no itching after 8 weeks. This was a 32 years old female non-diabetic patient. These are the results which proves itself that you need a scientific evidence. There is no medicine is available at the moment for psoriasis except steroids. Thank you so much.
Thank you,
And
Dr. Khan, for a very
one last suggestion for the future that anyone want or wish to translate its research to a clinical research, we are happy to collaborate.
Thank you for a comprehensive talk and also for keeping the time. Any questions, please?
Namko? Sir, is it possible to prepare its formulation as instead of oral dose?
We have already prepared the formulation. I have told you the nano suspension gel. We have applied the nano suspension gel for psoriasis patients. And we are going to patent it, so we have not added the data in the slides.
You think 8 weeks is a longer time?
What?
8 weeks is a longer time to heal.
We have, no, it's not only 8 weeks. There is a 3-year research. This is the only 8 weeks trial, which was been registered with the WHO. There are a lot of work have been done before.
So gel is based on fenugreek, is it?
Yeah, not only fenugreek, some more.
Yeah, but fenugreek.
But I have showed you the results.
Yeah, okay, thank you. Thank you very much, Dr. Khan. Now, please show your appreciation. Now we get on to the next speaker, who is online, Dr. Agusi, who is Assistant Professor in Biochemistry, Physiology and Pharmacology, Faculty of Science and Technology of the University of Abomey, Calavi in Benin. Oh, no, it's somebody else. Osama M. Ahmed. It is Agusi. Is he Abdullah? His name is not here. Shall we move on to the next speaker and come back? Huh? How about Abdullahi Ademi?
Hello, sir. Please, I'm having issue with my connection. I'll be presenting with KM Salawu.
Abdullahi, can I introduce you? This is Mr. Abdullahi Ademi Adegoke.
Yes, sir. I'll be presenting with KM Salawu.
Okay, go ahead. Something wrong with that. Here, there are two devices being used. Can you knock off one of them, either the phone or the computer?
Can they allow I'll be presenting on KM Salawu?
No, you are using two. You are connecting to Zoom with two devices. Can you knock one of them out? because it's disturbing. You can't hear what you are saying. Have you?
Assalamu alaikum. I said I will be connecting with KS Salahu username. I'm having issue with my connection on my laptop.
Okay, thank you. Go ahead.
Hello, I hope you can hear me.
We can hear you.
Yes, I say I'm having issue with my connection on my laptop. So I will be presenting with KS Salahu, KM Salahu username. Please kindly unmute so that I can present.
Please go ahead. You are muted. Hello. Shall we go next one? I'm sorry, we have to get on next one. We'll come back to you. Okay, the next speaker is Miss Rosina Parul of Gono Vishwavidyalaya, Nolam, Savar, Bangladesh. She will speak on.
Can you hear me?
Isolation. Oh, who is that now?
Can you hear me now?
Yeah, yeah. You
Oh.
are Muhammad Asma Khan. Is it? No, sorry. Ajwa say Agus. Abdullah Ademi. Okay, go ahead quickly.
Okay, thank you. Assalamu alaikum. I'm happy to be part of this conference to share our ongoing research work at the University of Ibadan. Although presently I'm a research student at HEJ ICCBS lab here, Karachi. The title of our research is in vitro anti-diabetic studies of different plant parts of 5 herbaceous species. Diabetes is a chronic metabolic disorder which affects everyone, and it is characterized by high level of glucose in the blood. And it is reversed to as 6 leading causes of death behind cancer, stroke, and others. And there has been a prediction that by 2030, 7.7% of world population will be diabetic. So that is huge. And about 1 in 9 of people, which account for about 589 million adults, are living with diabetes worldwide. And 1 in 8 of total adults with diabetes are predicted to rise to 853 million by 2050. And diabetes causes about 3 to 4 million deaths annually. And that is a record that has been set in 2024. And diabetes was responsible for an estimated 1.015 trillion in global health expenditure in 2024. And this represents about 338% increase over the past 70 years. So also 1 in 5 live births are affected by glycemia in pregnancy. How? In the world right now, there has been a sporadic increase in the number of diabetic patients all over the world. In 2024, about 588.7 million were diabetic, and there has been a prediction that it will rise to 852.5 million by 2050. Diabetes are classified into 2 majorly. We have the type 1 diabetes and type 2 diabetes. And the type 1 diabetes resulted from the insufficient insulin production, and where the pancreas cannot produce enough of insulin. And type 2 diabetes result from the insensitivity of the target organs to metabolize insulin.
You are muted. Unmute. You're muted. Can I unmute? You are muted.
Can you hear me?
Now okay, fine.
Okay. The current management for diabetes presently include dietary modification. As we all know, most of the glucose are derived from the daily consumption of our food. And there has been a recommendation to avoid this. We should stay away from consumption of food that are rich in rich in glucose and others. Then also exercise has also been the non-pharmacological management that have been prescribed, because through exercise, muscle utilizes cell and thereby increasing the sensitivity of insulin in the body. As well, oral antidiabetic drug has also been prescribed for the management of diabetes, which includes metformin.
Excuse me, your screen is not shared.
No, he's not. No slide.
He has no slide.
Yeah, no slide. No slide. He's talking, not no slide. Okay.
Can you see
Continue.
His computer is not working. Right. Go ahead, please. Yeah, only about 6 minutes more.
Can you see my slide?
Oh, your slides. You said something about your computer not working. You have no slides.
No, I've been sharing.
No, we are not
We
seeing.
cannot see your slide.
Oh.
I thought you said something about your computer not working.
Yes, I'm using a colleague's laptop so that I can make my presentation.
Anyway, you have only about 6 minutes more, so you better quickly do it.
Oh, okay. Can you see slide now?
Now, right.
Yes,
Okay.
we can see now.
Oh,
Thank
you. Okay. Better?
Okay, please.
All right. And
Go ahead to the present slide.
therapy that have been recommended. And this one is also majorly recommended for those with type 1 or type 2 diabetes.
Get on to your research work, please.
Okay. And with this, due to the side effects of these synthetic drugs, so the scientists have been looking for a way to maneuver the side effects such as flatulence, vomiting, and abdominal cramp that normally resulted from the use of synthetic drugs. And the Almighty Allah has told us in the Quran, chapter 16, verse 11, he said, He causes to grow for you thereby crops, olives, dates, palms, grapes, and all kinds of fruits. Indeed, in that is a sign for people to reflect. That means he's telling us that most of the diseases can be cured using...
Use slideshow because we can't see the slide you're talking about.
Is my slide showing now?
It's showing, but only one slide. It's not showing the slide you're talking about.
Oh. I don't know what's going on.
Now you have only 3 minutes left.
Is it showing?
No. Now right, yeah.
It's
But
showing up.
yeah, but please get your conclusions now because you have no time.
Okay. So the focus of this plan is to evaluate the different plant parts of Abyssal species for the management of diabetes. So the plants in the plant in focus is Abyssal lebec, Abyssal salmon, Abyssal ferrugini, Abyssal zigia, and Abyssal glaberrima. The objective is just to evaluate the alpha amylase and alpha glucosidase in vitro activity of these selected plant species, and also to compare their in vitro alpha amylase and alpha glucosidase of the.
I said to get to a conclusions, please.
Okay. The plants remain relevant to give novel remedy for mankind, but are simple. What's going on? Okay.
Yeah.
Oh, why is my slide moving like this? Can you see?
Yeah, I'll be a little bit. Okay, talk about it.
The voice is lost. What's going on? Can you see my slide now? Can you see my slide?
Yeah, we can read your slide. We have read it already. Get to the next slide.
So as I said, the work is ongoing, is currently ongoing. So from this study, we are able to conclude that the inhibitory activity of Abyssal lebec being promising to inhibit alpha amylase and alpha glucosidase were due to relative abundance of secondary metabolites like alkaloid, saponin, and flavonoid, and tannin. And this Abyssal lebec could be promising in the management of diabetes mellitus as it inhibited both alpha amylase alpha glucosidase in this study. So the further study to carry out include the fractionation of most active fruit extracts, bioactive guided isolation, structural elucidation of the isolated compound using spectroscopy techniques. So these are the exciting new and the exciting is about my research. So this research aims to bridge the gap between traditional knowledge and pharmacological application of this species, and also to offer a new bioactive compound from this species that will add to the repository of compound used for the management of diabetes, and also to develop patentable natural products for diabetes management, which will be beneficial to Africa and the world. So this is my selected references. Thank you for listening.
Thank you, Dr. Adegoke. Any questions? So there seems to be no questions. So thank you very much once again.
Thank you.
We'll get our next speaker, who will be Ms. Rosina Paro from Gono Vishwavidyalaya, Nolam, Thavar, Bangladesh. Miss Dr. Parul, please
Yes.
make a
Yes,
slideshow.
can you hear me? Assalamu alaikum.
Can you convert to slideshow, please?
Assalamu alaikum.
Yeah, fine. Go ahead.
And very good afternoon to everyone. Respected session chair, Professor Dr. Vijay Kumar, distinguished speakers, organizers, and participants. I am Dr. Rosina Parul, Assistant Professor and Head of the Department of Pharmacy, Gono University, Dhaka, Bangladesh. It is an honor to join you today. As a former under fellow, it is a privilege to present my research titled Isolation, Characterization and Computational Evaluation of Bioactive Compounds from Syzygium formosum leaves with Antidiabetic Potentials. Now, medicinal plants have played a vital role in human health since ancient times. Many modern pharmaceuticals have their origin in plant-derived active molecules, and diabetes mellitus is increasing globally, creating a demand for safer, more affordable therapeutic alternatives. Although cesium formosum is used traditionally for metabolic disorders, its anti-diabetic effects remain poorly investigated. This study therefore aims to provide scientific validation by evaluating the plant's anti-diabetic activity and identifying bioactive compounds responsible for glucose-lowering effects. Now, the objectives of our study was structured around four major objectives: phytochemical screening, isolation of compounds and their spectroscopic characterization, evaluation of anti-diabetic effects in steatosis-induced type 2 diabetic rats, computational analysis through molecular docking, especially targeting the SAR1 receptor. Together, these components allowed us to explore both experimental and computational dimensions of the plant's anti-diabetic potential as well. Now, we can see the methodology. This slide summarizes our integrated methodological approach. We followed a systematic natural product research workflow, beginning from extraction, fractionation, chromatographic separation, and structural elucidation, and extending to in vivo anti-diabetic evaluation in steatosis-induced diabetic rats. And the combined in vivo, in vitro, and in silico design strengthen the validity of our findings. Here I am presenting the experimental design for our in vivo study on steatosis-induced type 2 diabetic rats. After inducing diabetes with steatosis, the study was divided into an acute phase and a chronic phase. In the acute phase, we conducted OGTT on day 0 and day 14, collecting tail tip blood at multiple time points to measure immediate changes in serum glucose. And in the chronic phase, which lasted 28 days, the rats were assigned to three groups: a water control group, a glycoside standard drug group, and a group treated with 80% ethanol extract of cesium formosum. And during this period, we monitored weekly body weight and collected blood on days 0 and 14. On the day 28, the animals were sacrificed and we collected blood from the heart and liver tissues as well. These samples were used to assess serum glucose, lipid profiles, and liver glycogen levels. And overall, this design allowed us to evaluate both the short-term and long-term anti-diabetic effects of the plant extract compared to the standard drug treatment. Now we can see the some isolated compounds from these plants through chromatographic techniques and spectroscopic characterization. We successfully isolated and identified these compounds from leaves of cesium formosum. Here, the docking study overview for the computational component, we selected the SAR1 receptor, PDB ID 6PZI, which plays a key role in insulin secretion pathways. Glycoside, a standard sulfonylurea drug, served as our reference ligand with a docking score of 8-8.3. The docking results showed promising binding affinity. Several isolated compounds demonstrate notable interaction with SAR1, some approaching the affinity of glycoside, and key binding features including hydrogen bonding, hydrophobic contacts, and interactions with crucial residues that regulate insulin release. And these observations suggest that the compound may act through insulin secretory-like mechanism. Here we can see the in vivo study design. By this, we validated the activity in steatosis-induced type 2 diabetic rats. The 80% ethanolic extract was administered at a dose 1.25 gram per kg. We observed significant reduction in blood glucose, increased liver glycogen storage, and partial regeneration of pancreatic beta cells. And standard biochemical and histopathological methods were applied to ensure accuracy and reproducibility. From these photographs, visually represents key steps in our in vivo experiments: steatosis-induced injection for inducing type 2 diabetes, and tail tip blood collection after minor amputation, centrifugation for serum separation, and these images ensure transparency and reproducibility of our experimental model. Now, you can see the OGTT results show that all groups had similar fasting glucose levels, but all experienced significant increases following glucose loading. One way ANOVA revealed no significant differences between treatments, indicating the glycoside and cesium formosum did not improve glucose tolerance. All groups demonstrate impaired glucose regulation consistent with type 2 diabetes. Now, this slide shows the body weight changes of rates across 28 days in all 4 groups. The weights remained relatively stable over time with no significant differences between the groups at any time point as confirmed by the one-way ANOVA. And these findings indicate that none of the treatments caused weight loss or weight gain, suggesting they had no adverse or beneficial effect on body weight during the study period. This slide represents the fasting serum glucose level at baseline, day 21 and day 28 across the experimental groups. One-way ANOVA revealed no statistically significant differences between groups at any time point, indicating that the treatments did not produce group-wise alteration in fasting glycemia. However, pair t-test analysis demonstrated significant within group reduction in the glycoside and cesium formosome cohorts by day 28. Suggesting a treatment-associated improvement in fasting glucose regulation over time. From this slide represents the 28-day lipid profile outcomes. Although the one-way ANOVA shows no significant differences between groups, the pair t-test reveal a significant reduction in triglyceride in the cesium formosome group and modest within group improvements in glycoside. These findings indicate that while group-wise effects were limited, certain treatments exerted meaningful lipid modulating effects over time. This is the graphical representation. And from this slide, across all lipid parameters, including triglyceride, cholesterol, HDL, LDL, and the respective ratios between group differences were not statistically significant. However, paired analysis revealed meaningful within group improvements, particularly in the glycoside, cesium formosome groups. These trends suggest potential lipid modulating effects despite the absence of a strong group-wise statistical separation. This is the graphical presentation. And this slide shows the liver glycogen content was markedly elevated in the glycoside group, whereas the cesium formosome group exhibited a modest increase to 109% relative to the normal control. And although the cesium formosome induced elevation did not reach the magnitude observed in glycoside, the statistical comparison indicated that cesium formosome exerted a measurable but comparatively weaker enhancement of hepatic glycogen storage. And this finding suggests that cesium formosome has a mild positive influence on hepatic glucose reserve capacity as well. Here we can see suggests that mechanistic insight that suggests that the struct likely adds to SAR1 mediated insulin secretagogue activity and thereby supporting its overall hypoglycemic effect. Now, in summary, this study provides the first comprehensive scientific report on cesium formosome, including the successful isolation of novel bioactive compounds. The compound demonstrated strong docking affinity toward key antidiabetic targets, and our in vivo findings further validated its therapeutic potential. Collectively, these results position cesium formosome as a promising candidate for future antidiabetic drug development. The future perspectives moving forward, structure analog synthesis and molecular dynamic simulations will help refine and validate the mechanistic basis of the identified compound. Comprehensive pharmacokinetic and toxicity profiling will be essential to establish its safety and translational potential. Additionally, expanded biopsy-guided purification may uncover further active constituents, strengthening cesium formosome's promise as an antidiabetic source. Now, I would like to express my sincere gratitude to ANDRA, Gonu University, BUSS Department of Chemistry and Pharmacology, and Jahangir University for providing continuous academic and laboratory support throughout this research. I am also deeply thankful to all collaborators whose expertise and cooperation significantly strengthen the quality of this work. Their collective contributions were essential to completing this study. Thank you, all of your kind attention.
Thank you, Dr. Rosina, for your clear presentation and for keeping to time. Are there any questions?
We have some online questions also, sir. Let me first take it.
There is nothing I can see as a question.
The question that is by Noman Khan. That question.
Which one?
Noman Khan. The question is by.
No man, that's for Dr. Osama. That's not someone who spoke today.
He's asking the question, have you used a single concentration of fruit and seed?
Who is Dr. Osama?
Yes. But he's already did it. Yes.
Okay, Dr. Osama, have you used only a single kind of fruit and seed? But Dr. Osama didn't make a presentation today.
Okay. So the next one that is by Dilawar.
No, that's it.
Is the one question.
Who is a question for whom? How do you evaluate the drug presence by docking? Is there any in vivo or in vitro method involved? This is for Rosina. Can you answer a question, please?
Yes, of course, it is a docking method, which was done by some softwares, as I mentioned in my presentation.
Dilawar, does that answer your question?
Yes, here we use a protein structure of a standard antidiabetic drug, sulfonylurea, that is indicated SAR1, which is collected from PDB, protein database bank, and after that we use some. And here I shown the ligand and as well as binding affinity of my isolated as well as which is compared with SAR1 and shows this consistent binding affinity, and that is already presented.
Thank you, Dr. Rosina. With that,
Yes.
we have no more questions for you. Thank you very much. Show appreciation again. The last speaker for the day, because we couldn't make contact with Dr. Agusu of Benin. So last speaker will be not online, he will be speaking here, and that is Professor Dr. Ziauddin from the Department of Pharmacy, Comsats University, Abbottabad campus, Pakistan, and he will be speaking about exploration of the antibiotic potential of Buxus wallichiana, bridging traditional use with pharmacological evidence. Dr. Udin, you have 20 minutes.
Bismillah ar-Rahman ar-Rahim. First of all, thanks to all the audience for waiting till this time. And I am also thankful to the chair for the introductions. So being a last speaker, I will go straight to my work, which has been done by my research students. So the title is Exploration of the Anti-Diabetic Potential of Buxus wallichiana, which is a traditional plant in Pakistan, especially in the northern areas, and bridging the traditional use with the scientific exploration. I will skip what is diabetes, its types, and other statistics, but fewer I will present here, like the global burden of diabetes, 589 million people are suffering. And mostly the type 2 diabetes, and most of the cases, like more than 75%, have been reported in the low and middle income countries. So especially in Pakistan, the third largest diabetic population in the world, and the prevalence in Pakistan is 31% in the adult population. So it makes 34.5 million of adult Pakistani from age 20 to 79, they are suffering from diabetes. So over here we can see then the 1 out of 4, they are undiagnosed, and 1 out of 10, the adult, they are suffering from diabetes. So different treatment strategies are adopted, but there are some traditional plants which are used by the people, the local people in the hilly areas, but there is no scientific evidence that whether they have some scientific chemicals or some things that can be used. So in this regard, we have chosen this plant, Buxus wallichiana, which is used in Malakand division of the northern Pakistan, especially in the district Bunair. The people are using for the diabetes complications, but they don't know scientifically. And when we searched the data, the literature, there was no evidence. So we surveyed the local community, collected the plants, and then tried to investigate the in vitro and the in vivo activity. So the natural habitat, the northern areas of Pakistan, it's a shrub having more than 70 species. And traditionally it has been used for various ailments, especially in diabetes. So this is the experimental design. The work has been done by my grad students like MS and PhD, collection and identification, processing, chromatographic analysis, phytochemical analysis, the in vitro and in vivo investigation. So the plant was identified and the voucher number was given by a taxonomist in University of Swat. Then the collection, drying, and the processing, filtration, and extraction. So first of all, we did the in vivo study using the bulb C mice, and these are the material methods, and some I have mentioned here. But I will go straight to the results and discussion. Like here we can see the fresh plants, we have used the leaves, 8 kg of the leaves were collected. And then when they dried, so the weight became 1 kg, and then we extracted, the extract was 80 grams. And the extract was then checked for the phytochemical analysis. Here we can see that which class of compounds are present, the flavonoids, alkaloids, and glyphosate and saponin, they were present, while the tannins were not found. Then we did the TLC of the crude extract, and here we can see that multiple compounds have been reported, which are shown here. This is the HPLC, a different wavelength 254 and 365. So the initial alpha glucosidase enzymes and the PTB1B, which are the two enzymes causing the diabetes, they are available in our lab. So we also designed our study targeting these two enzymes. And we calculated the IC50 value of the crude extracts, and it is shown here like the 2.54 microgram per mole is the IC50 value targeting the alpha glucosidase enzyme. After that, we also performed the PTP1B inhibitory activity of the crude extract. And here it's shown in the table, like the 1.45 microgram per ml is the anti-PTP1B inhibitory activity. So it means that the crude extract strongly inhibited both of the enzymes. And if we compare the alpha glucosidase and the PTP1B, so they showed potent inhibitory activity against the PTP1B. After that, we used the in vivo model mice model, and 5 groups like the normal control, diabetic control, the positive control, and the 2 groups of the extract. So here we can see the body weight, the effect of the crude extract on the body weight of the mice. So in the normal control, there is no change, but in the diabetic control, there is a drastic decrease in the body weight of the mice. But when we compare with the glibenclamide, which is the positive control, there is increase in the body weight. And when we see the 2 doses like the 500 mg per kg and the 1000 mg per kg, there is also a slight increase in the body weight. So it means that they have some anti-diabetic activity. Furthermore, we checked the glucose level. This protocol was adopted for 14 days. So on day 0, day 2, day 4, day 6, day 8, day 10, day 12, and 14. And if we compare the treated group with the negative control, there is decrease in the blood glucose level, especially on day 14. After that, the blood samples were collected, and the triglyceride, cholesterol, LDL, and HDL level of the blood was also monitored. So here in the figure, it is evident that the treated group decreased the blood cholesterol level as compared to the negative control. Although our results are not significant in comparison with the glibenclamide, which is a standard drug, but when we compare the results
with the negative control. So it is evident that there is decline in the blood glucose level, the blood cholesterol level. Similarly, in the triglyceride, here, there is a slight decrease in the blood triglyceride level of the treated group as compared to the negative control. The HDL level, the high density leveled protein. So here is a slight increase when we compare with the negative control. So both the treated groups like the 500 mg and 1000 mg, they have increased the HDL level. And the last one is the LDL level. The LDL level has been decreased as compared to the negative control. While the 500 mg, the effect is little one, but the 1000 mg per kg dose has significant effect in lowering the LDL level. So in conclusion, the phytochemical analysis of the crude extract of Bacillus willichiana have revealed that flavonoid, glycosides, and alkaloids are present. The crude extract displayed in vitro antidiabetic activity by inhibiting the alpha glucosidase and PTB1B inhibitory enzyme by IC50 value 2.45 and 1.45 microgram per ml respectively. The in vivo study also displayed that the crude extract has significantly decreased the blood glucose level as well as the cholesterol, triglyceride, and LDL level. These are some of the references. And acknowledgement, I would like to acknowledge the grant of IFS, with the help of which we have established a new lab and trained students, MS and PhD, and have provided the opportunity to work, and the Comstech University and my students who have done this work. Thank you all.
Thank you, Professor Udin, and thank you very much for saving us a lot of time. So we have a lot of time for questions. Yes. Dr. Khan.
Thank you so much for your nice presentation. In your presentation, you showed a slide of HPLC. Would you please let me know which standards you have used for the HPLC chromatogram, and if you have validated the sample, which QC sample and control 0 you have used?
Thank you. It's a very interesting question. Actually, this is preliminary work. Even this work is now going on. We are doing the isolation of the pure compounds. We have isolated 4 and 5 compounds, and now we are doing the NMR. This is not completed yet. Regarding the HPLC, we have just prepared the crude extract, develop a method, run without any standard. We didn't run any standard. This is just the initial preliminary HPLC. Now we are going to isolate each peak, like the peak which are presented at HPLC, they mean they're UV active peak. But we have isolated some UV inactive compound as well after staining the TLC plates. So we didn't run any standard with this. This is the only pure peaks of the extract, and now we are going to isolate the compounds.
Thank you. Any other questions?
I have. Thank you very much for your very nice presentation, but I have queries. You use the dose of cryptozotocin 120 milligram per kg body weight. That's really a very big dose. Then what is your level of glucose when you check them as hyperglycemic or diabetic? This is one question. Okay, then I have a series of questions. Just to clarify, how did you work? Then you said that you collected the blood from the mice every second day, 4th day, 6th day, 8th day, 10th, 14th day. What is the necessity of doing that? You cannot do this stress to the animals which are diabetic. No, never. Our ethical animal ethics is very strong. It never allows to check only the condition how this diabetes is reducing with the help of your plants. It's not the way. you know? You have to take after 1 week. Still, it is not allowed. You have to give them some time to regenerate or to work. And you know, by collecting the blood. Okay, you did, I think, by strip method, yeah? Okay. For research, also it is not allowed to do always to doing the strip, because we know the peripheral blood, when you are doing with the strip method, is not feature the exact value for glucose. So I think it would be better if you started at the 0 day, then at the 14. So there is a very big gap, and you could see the same result if you're getting the gradual decrease of blood glucose. So this is my comment, but just I wonder why you use that such a high dose. Actually, starting from the dose 50 milligram per kg body weight and up, you use 120 milligram per kg, but it will destroy all the pancreatic beta cells, and they will give you a model which will mimic type 1 diabetes. And to you, when all the pancreatic beta cells are died or dead, then how your plant extract will help to produce insulin to help to reduce the blood. So this is my question. I don't know I wanted to know the initial rise of blood glucose. What was before injecting streptozotocin, and what came after immediately after injecting streptozotocin? Because you have to have the baseline starting the experiment, then you inject, then after sometimes you have to wait. Usually for type 1, we use for 7 days. We have to wait, and then we go for checking the blood glucose. For type 2, it's absolutely different. The dose is different. The model, the methodology, there are lots of methodologies of producing type 2. So I wonder, what was your type of diabetes you produced?
Yeah, thank you for suggestion and comments.
Yeah.
Actually, it was type 2 diabetes. And another thing is like the streptozotocin dose, we can start from 50, and we have followed the procedure of published data. So we followed that procedure. And regarding your question of the multiple time fracture or getting the blood samples, so we have also followed the reported method. Yeah, I agree with you that there are.
Make actually no sense, because you are doing stress, and animal ethics research never allow you to do that, because the animal is getting stressed. So we are not allowed to make that. So what is your aim? You want to see that your plant extract is lowering the glucose. So you weigh, okay? You just induce, you're confirmed that your rat or mice is diabetic. Then you feed, and then you wait. You may giving them continuously feeding part every day, and then you can wait up to 7 days at least, but not, please, not customary to take so many times. It will be ridiculous. You will get, due to stress, the sugar level may rise, okay? That won't depict the real picture of your extract. Your extract may be very good, but you are taking the stress to the animal, okay? So then next, I think your plant is very good. It actually shows the hypoglycemic activity, but the method you followed, you should a little bit wiser to use that. Okay.
Thank you.
The time for one more question if you want, everybody. Yes.
Assalamualaikum. Have you performed your toxicity studies before selecting doses?
Yes, we have performed, but I didn't mention here. According to OECD guidelines, we have used multiple doses like 200 mg, 400 mg, and 800 mg. And during these doses, we didn't see any lethal effect like salivation, lacrimation, and even death of these dogs.
And do you experiment any animal die because you are using a high dose?
No, we didn't observe any death of the animal.
Okay, thank you very much, Professor Udin. Thank you for a very good lecture which provoked so many questions.
Thank you.
Thank you. And it's my duty now to thank all the speakers, We had 5 speakers, but unfortunately we could not make contact with Dr. Agosu of Benin. So I would, on behalf of all of you, thank Dr. Khan, Mr. Adegoke, Dr. Parul, and Professor Udin for their wonderful lectures and for having kept to time. Thank you very much. In 5 minutes, we start the next part of the program.
Okay, thank you very much. Please appreciate our chair for the technical session. Okay, so we'll just begin with the closing ceremony of this 2 days NRAB and comstech regional seminar for the year 2025, plant materials and their clinical validation in the management of diabetes and metabolic disorders, their challenges and scope. So for the closing ceremony, may I request Professor Dr. Begum Ruqaiya, Network Leader and Secretary General of NRAB and Dean Bangladesh University of Health Sciences for her closing remarks on behalf of NRAB.
Assalamu alaikum, everyone. We have come to an end of this 2 days seminar, 8th regional UNRAP conflict seminar on plant materials and their clinical validation in the management of diabetes and metabolic disorders, challenges and scope. So I think during these 2 days, we have learned a lot because there were eminent scientists and researchers from many countries. If I make a note, there were presentations from Bangladesh, from where I am. Then we had a presentation from Sri Lanka. We had presentation from Australia, Pakistan, Egypt, Nigeria, United Kingdom, and there supposed to be one presentation from Benin in the last session, but he was supposed to give his lecture online, but we could not contact, but otherwise his abstract was there. So, ladies and gentlemen, I think we had gone through this 2 days seminar and learned a few things. What I wanted to mention that natural products, we should value them because natural products, sometimes we say that they are as they are natural, so they are safe. So this is not always true. We have been using natural products as remedy of various diseases since ancient times. But this is our duty in modern era to scientifically evaluate their medicinal value, to say people that this is safe or not, or this is working or not. But we heard that during these 2 days that more than 80% of the population of the developing countries rely on plant materials as their primary health care. So it is very much important and very much imperative for us to scientifically evaluate the plants. And for diabetes, it is very much needful because we heard in every lecture that the diabetes is increasing in an epidemic rate. And as International Diabetes Federation already predicted that by the year 2050, there will be a 75% increase in diabetes. population. And this picture is very much furious in terms of the Southeast Asian region where we people reside. So now I think it is our duty to actually test those plants that have folkloric reputation, or our traditional healers use them for the treatment of diabetes. So with this note, I want to make a conclusion that all the papers that were presented here were very scientifically sound. Everyone tried their best to present, and of course, did very hard to they did hard work to deliver good lectures. So I do believe that we actually understood the importance of natural products, importance of plant materials as source of remedy for various diseases, especially for diabetes. So I would like to conclude this session by giving the by giving the certificates to the participants. And I also want to mention that in our inaugural session, we were delighted to have the minister from Ministry of Health of Pakistan, and we had our from Bangladesh Embassy. There were representation from Bangladesh Embassy, his High Commissioner of Bangladesh also present, and so many distinguished people came to this meeting to encourage us, and we are grateful to them. And I would like to express my sincere gratitude to you all, because without you, without your presence, it could not be successful. So thank you very much, the audience. I'm really, really grateful. And I also want to extend my sincere gratitude to the OIC Comstech Secretariat, specifically Khazima and her team, because all the time we have been corresponding to make this seminar a success. You know, without their assistance, we could not make this seminar a success. I don't know whether it has become successful or not, but I do believe that we tried our best with the OIC Comstech Secretariat. So let us have a big hand to the Comstech Secretariat. What else? Thank you very much, ladies and gentlemen. And I think we will come to the end of the ceremony for the presentators will get the certificate. Okay. Yeah. Oh, okay. Okay. Okay.
Thank you very much, Madam, for your kind words. Ladies and gentlemen, His Excellency Professor Dr. Iqbal Chaudhry is being represented by Professor Dr. Shahid Mahmood, who is Senior Advisor at OIC Comstech. So I'll request him to please come and say a few remarks on behalf of His Excellency. Please welcome him.
Assalamu alaikum, and a very good afternoon. It is indeed an honor to be here on behalf of His Excellency Professor Dr. Iqbal Chaudhry. I am honored to start by recognizing Professor Dr. Begum Ruqayya, and with her the national Dr. A.K. Azad Khan, Professor Wajia, Professor Nasir, and Professor Atia. It is something that I was, while sitting and listening to you, Dr. Ruqayya, reminded me of being back when I was doing my PhD and you went out and did research. In fact, from the heart that you spoke, trying to guide the gentleman who was presenting, took me back to the feeling of what it is like. As we go along, we get more into the administrative work. And I'm glad to see that this academic slash sharing of knowledge session very ably being guided by yourself and the team who has participated and been here as presence and speaking. It is indeed an honor for OIC Comstech to be hosting this. It is also very, very important at this juncture that such a seminar was organized, because as we are moving along, we are quite clear that because of the way the world is changing, because of the climate impact and the way we are trying to balance health, especially diabetes as it was being addressed here, there are a lot of challenges. As much as knowledge sharing and experiences are passed on amongst each other, and the knowledge base from what we know as possibilities using plants, and especially to a menace like diabetes, is critical. And it is heartwarming to see such a gathering. It is heartwarming to see such participation. And it is also something that is timely. OIC Comstech, under the able guidance of Dr. Iqbal, is trying to organize and maximize the exchange of science and technology knowledge across the OIC countries and those countries that participate with us as technical partners. I am, on his behalf, truly grateful to all of you for being here and honoring us by the presence and the sharing of knowledge. And especially Professor Ruqayyah, it is your able guidance and all the senior professors that have made this possible. Thank you very much.
Thank you very much, sir. Now I request Professor Shahid Mahmood and Professor Ruqayyah to please go downstairs to distribute certificates of appreciation to our speakers first. So first we have Yes. So the first one, I would like to invite Dr. Nasar Ahmed. Sir, thank you very much for your contribution as a speaker. Professor Dr. Vijaya Kumar. Thank you very much, sir. Professor Dr. Shamsun Nihar Khan. Thank you, madam. Professor Professor Dr. Atiyatul Wahab, please appreciate our speakers who have spent their time to share their work. Dr. Ranzu Ahmed from Bangladesh. Dr. Rosen Zamir from Bangladesh. Dr. Ziauddin, please come and receive your certificate. Dr. Muhammad Asghar Khan. For the oral presentation, Mr. Mir Tofkir, what is it? Mr. Mir Rahman, please. Mr. Ashrafuz Zaman, please receive your certificate for the oral presentation. Okay, so with this, we come to end of the certification distribution ceremony. So, sir and madam, thank you very much. And for all the participants, you will receive your certificates from us, from the comstech team. We have arranged a tea outside for our participants. Thank you very much, and thank you for your time. Yes, and for the virtual participant, you will receive your e-certificates. Thank you very much for joining.